Okay, well, I'll do a little introduction maybe to start. Welcome everyone to the trait evolution section in the virtual Asilomar meeting. Thanks to everyone for joining us. We have an interesting slate of speakers today, 20 minutes each. Most talks are being given live, a couple pre-recorded ones with, I think the attendees may be here to answer questions as well. So we'll get things rolling right off to give everyone their allotted time. Hopefully things go smoothly on Zoom. Please remember to keep yourself muted. Questions are submitted and chosen by our question moderator. I don't think there's anything else we had to cover, so I'll start by introducing our first speaker, Robert Colwell, who's going to be telling us about adaptive trade-offs that are concealed by allometry among hummingbirds. So I'll turn it over to Robert.
Thank you, Howard. These are, whoops, one moment. These are my amazing co-authors, each of whom made crucial contributions to this project. We aim to submit the underlying manuscript to the American Naturalist shortly. In fact, this study was originally inspired and stimulated by fieldwork I did exactly 50 years ago and published in MNat in 1973. When most people think about a hummingbird feeding on a flower, they envision the bird hovering with its bill legitimately inserted in the flower, extracting nectar and picking up or delivering pollen. We call this the orthodox feeding style. Hummingbird bills vary tenfold in length from 8 millimeters to 8 centimeters. These birds with very short or even very long bills can feed in this way, and all hummingbird species do so at least part of the time. But some species of hummingbirds have unorthodox feeding styles that they use often. These three are clinging with their very impressive feet to leaves or flowers in a vertical position while feeding legitimately through the corolla opening and potentially delivering or picking up pollen. Clinging to feed legitimately is apparently more difficult the longer your bill, so these are fairly short-billed species. This acrobat, a high elevation species that often walks on the ground, has the large feet it takes to cling even upside down while feeding legitimately on short corolla flowers with its short bill. The two species of Eutoxeres are the rule breakers. They are nearly obligate clingers, but they have long bills. However, the bill is strongly decurved to reach around the corner into the curved corolla flowers, like this heliconia, that they feed on legitimately and pollinate. Among the hummingbird species that are capable of clinging, some are nectar robbers. The two on the left are clinging to the flowers themselves. The one on the right is clinging to the inflorescence. They tend to have shorter bills. These species feed illegitimately without picking up or delivering pollen. They extract nectar by piercing the base of the corolla or through existing openings, either natural openings between petals or existing pierces made earlier by themselves or by other species of robbers, such as the passerine birds called flower piercers, genus Diglossa. Another category of unorthodox feeders are the hovering nectar thieves. They pierce corolla bases or feed through existing openings on the wing. Because they need not cling to feed, bill length is not much of an issue. Bills of species known to feed in this way range from 11 to 28 millimeters. The final category of unorthodox feeding styles comprises a small group of high elevation Andean hummingbirds that feed from the ground legitimately on cushion plants, either perching on the ground like the bird on the left or clinging to vertical surfaces like the one on the right. All hummingbirds perch to rest, and most will take advantage of a convenient perch to feed legitimately, as everyone who has ever had a hummingbird feeder can affirm. But perching is not clinging in the technical sense that we use the word in this study. All hummingbirds hover to feed, but not all cling. Has clinging behavior originated multiple times in hummingbird phylogenetic history? What does functional morphology predict about clinging? Do the feet of clingers differ from the feet of non-clingers? Do the bills of clingers differ from the bills of non-clingers? To answer these questions, we collected both behavioral and morphological data. that behavioral data came from several sources. First of all, the published literature, but also every hummingbird photo and video in the online version of Del Hoyo's Handbook of the Birds of the World. And finally, field notes of the authors and other informants. Of the 220 species for which we have morphological data, 76 are known to be unorthodox feeders in one way or another. Of those 66 species are known clingers. We built 1,000 phylogenies for the 220 species in our study based on Jimmy McGuire's genetic sequence data. We mapped all known unorthodox feeders on a modified consensus tree and applied a simple parsimony analysis to estimate the number of independent origins of unorthodox feeding styles. Here's the phylogeny for the 220 species of hummingbirds with unorthodox feeding styles indicated at the species tips using the shape and color codes shown here. Each inferred independent origin of clinging is marked with a yellow star with clinging lineages marked in yellow. Here you see 9 origins just within the emerald clade. Look for the little yellow asterisks. But there are many more in the other 7 clades of hummingbirds. By far the most ancient origin of clinging behavior lies at the base of the coquette clade, now scrolling past. You see over and over an independent origin. Based on current knowledge, there have been 24 independent origins of clinging to feed. Clinging has arisen at least twice in each of the 9 clades, except for the monospecific clades Patagonia, a clinger, and Florisuga mellifera, a non-clinger. As we saw, clinging has a basal origin in the coquette clade. What about undocumented clingers lurking among the non-clingers? What effect would they have on this analysis? A lurker is presumed, in a presumed non-clinger clade will add an origin. A lurker, congeners of known clingers will neither add nor subtract any origins if they imply a common origin. Finally, lurkers that link 2 current origins by parsimony will subtract an origin. In short, there is strong evidence for multiple independent origins of clinging. Here's a video of an Amazilia in an experimental setup, clinging to a substrate. The reality of clinging in hummingbirds is far more complex than our treatment here, considering the basic construction of the hind limb apparatus, but here are the basics. Gravity enacts moments of torques about the joints of the leg, toes, and claws, tending to pull the bird off the substrate. The muscles of the distal hind limb must generate forces to counteract the force of gravity and balance the torques. We cannot obtain accurate measurements of all these torques for 220 species, so we simplify the analysis to 2 key measurements we have obtained for all specimens, tarsus length and hallux claw cord. The effect of tarsus length on clinging ability is analyzed here. Based on fundamental principles of biomechanics of clinging as described for other species, our initial hypothesis was that clinging species should possess shorter tarsi than non-clingers to reduce the moment of gravity. A shorter tarsus should offer a mechanical advantage for a clinging hummingbird. A longer one gives a disadvantage. As for hallux claw, a shorter claw requires a clinging hummingbird to expend more work to cling. A longer hallux claw requires less work to cling. So the predictions from first principles of functional morphology are, first of all, that clingers should have shorter tarsi than non-clingers, and they should have longer hallux claws than non-clingers. To assess these predictions, we measured more than 1,600 museum specimens of 220 hummingbird species out of the approximately 340 species currently recognized. The features that we measured were tarsus, hallux claw cord, middle toe claw cord, and bill length. And then mostly from the literature, but from specimen leg tags when necessary, we recorded body mass as live weight in grams. 3 factors complicate morphological comparisons among species. First of all, phylogenetic autocorrelation, and of course, body size and allometry, and finally, elevation above sea level. To neutralize these confounding factors for allometric analyses, we used PGLS to filter out the effects of phylogeny. We refer to these data as phylo-filtered data. For comparing or correlating the size of foot features and bill length in clingers versus non-clingers, we used PGLS to filter out the effects of phylogeny, body size, and elevation. We call these triple filtered data. To account for phylogenetic uncertainty, we applied the method of co-author Gonçalo. Statistical methods we applied included simple t-tests with Hedges G to assess the effect size, and standardized major axis with R² for bivariate relations. All morphological variables were transformed before PGLS, and filtered morphological variables for each species were post-GLS means from 100 alternative phylogenies. We assessed the leverage of morphological outliers. So here are the results for allometry for bill, tarsus, hallux claw, and middle toe claw. All four are positively allometric, although middle toe claw in the lower right differs only slightly from isometry, what you would get from a photographic enlargement. For bill length, the slope is considerably allometric, and clingers and non-clingers differ significantly. The clinger line lies significantly above the non-clinger line, so a clinger for the same body size has shorter bill. I said that backwards. The clinger line significantly above the non-clinger line. So they have, that was backwards. Sorry about that. That's a mistake. Should be the other way. Anyway, clinger allometry does not differ significantly from non-clinger allometry for foot variables, tarsus, hallux claw, or middle toe claw. Here are the univariate comparisons of bill and foot element size, and these make very clear what the differences are. Clingers have very significantly shorter bills than non-clingers, which is what I meant to say in the other slide. And they have longer hallux claws than non-clingers, also highly significant. Neither tarsus nor middle toe claw differ significantly between clingers and non-clingers. Here we see a crucial plot which reveals the underlying negative correlation between bill length and hallux claw length, foreshadowed by the photographs of feeding styles at the beginning of my talk. The fuzziness is the intentional result of overlaying the same plot for 100 alternative phylogenies to illustrate the very modest level of uncertainty due to alternative phylogenetic reconstruction. Each of the 220 solid points colored to indicate feeding style shows the bivariate mean of 100 triple filtered variates for a single species. So there are 220 of those solid points. The 100 superimposed green lines show the negative SMA relation between bill length and foot size advertised in the title of this talk. The inset shows the mean values for each feeding style on the left. All the means for the clingers lie in the lower right quadrant of the screen, which means short bills and long hallux claws, as we saw from the univariate results. Here we diagram the correlation structure among the four key morphological variables. The only correlation that strongly unites clingers is the negative one between bill and hallux claw. The intercorrelation among the clinger foot variables, tarsus, hallux claw, and mid-toe claw, is weak. In contrast, positive intercorrelation among foot variables and the bill is strong among non-clingers. So the conclusions are that the prediction that clingers should have shorter tarsi than non-clingers was not confirmed. We conclude instead that longer hallux claws are the key morphological innovation of the foot that enables clinging to feed by expanding the grasp of the foot. Hummingbirds display a surprising but functionally interpretable trade-off. And for trade-off, I simply mean the sacrifice of one benefit for another. Short-billed species repeatedly evolved exceptionally long hallux claws. They sacrifice the benefit of legitimate access to abundant nectar in long corolla flowers, but their biomechanically enhanced feet offer two compensatory benefits: saving energy by clinging to feed legitimately on short corolla flowers and stealing nectar from long corolla flowers. Thank you for coming to my talk. Here's an acknowledgement.
Great, I'll be here to relay any questions if people want to throw those in the chat or the Slack. I don't see anything quite yet, so I'll start with my question, which is, so it seems that although clingers have generally shorter bills and things like this, there's a lot of scatter in the data, and so these aren't hard constraints. And you kind of pose at the end there that, you know, the trade-offs, which I was wondering about. Given clingers are in the minority, do you think those costs that you discussed in your conclusions kind of outweigh the benefits?
Well, given that it's arisen many times and has apparently been lost sometimes within the coquette clade, which had a very early origin of clinging, it seems to be very context dependent. And the context may involve which other hummingbirds have overlapping geographic ranges, and of course, which flowering plants have flowers that are appropriate for short bills, long bills, piercing, and so on, within the range of a particular lineage. So it seems to be rather a plastic character. The most plastic character in hummingbirds is probably the bill, which goes all over the place. And you can have one species where male and female have very different bills or similar bills despite different body sizes and so on. Does that help at all?
Yeah, that also, yeah, gets at that variation in general.
Yes, there's all that scatter in that bivariate plot is not noise, that's music, that's evolutionary music.
Yeah, I completely agree. I just wonder if some hummingbirds are, you know, having it all with their long bills and access to everything. But I'll move on to Scott Powell's question, which is, with so many origins of clinging, do you have any insights on what might be driving the switch, habitat, degree of flower generalization versus specialization? You touched on that a little bit, but.
Yeah, I think it's a biogeographic context, and it's going to be extremely complicated because we can't just take the flowers as a given or the birds as a given. I mean, they're co-evolving with each other in a very diffuse way. Plus the geographical situation. We have one case where I was puzzled by Eugenes fulgens being an absolute non-clinger, just a long-billed bird that fed normally on long flowers in Costa Rica. Then I got some evidence from Mexico that it's a clinger. Well, it turns out they're different species. It was just a paper published last fall showing that they're different species. So I'm dying to know if the toenails are different in the Mexican one than the Costa Rican one. So even within sister species that were thought to be one species till October, there could be differences. I think it's, all I can say is it's got to be very context dependent.
Okay, and I'm going to follow up with a portion of Anurag Agral's question, which is that I'm curious about. Are there other traits that aren't measured in this study that you think go along with these traits?
We tried to measure everything we thought was relevant. We also measured wing length, but that doesn't seem to be particularly important. And you have to, when you're using museum specimens, you have to measure something that doesn't change length when it's dead and dried. So that's why we ended up using the claws of the toes rather than the toes themselves, for example. And the tarsometatarsus, or the tarsus, is a stiff bone, so that works okay. And of course, bills are stiff, but the muscles and tendons that are involved in this clinging stuff are just extremely difficult. You can't do anything with dead specimens. You'd have to do dissections or experiments or something, which we haven't done.
And kind of a related, on the flip side, question from Jimmy DeMeo is kind of, are there other proposed functions that aren't analyzed in the study that longer hallux claws may be useful for?
Yes, actually, in our written version of this, we'll discuss the use of claws for grooming. And there's some thought that a long-billed bird has to use its toenails for grooming because it can't groom with its bill because the bill is too long to reach the back of the neck. So there are complications like that. And another thing I just remembered is that you noticed in one of the birds I showed that was a high elevation bird that it was propping itself up with its tail. And so there are some, especially in the coquette clade, birds that use their tail while they're clinging. And this, of course, changes the functional analysis of stresses and counter moves by the bird's muscles and so on. So that's another complication, but just within that one clade.
That's great.
By the way, we looked at this same analysis within the coquettes, and the same thing is true. Even just within that one clade, the clingers have longer toes, longer toenails, and shorter bills than the non-clingers.
Great. Thanks, Robert. We're going to have to wrap it up there to keep on schedule. Appreciate it.
Thanks so much.
Our next speaker is Matthew Nielsen, who's going to be talking about climate change associated evolution of reaction norms. So I'll turn it over to Matthew.
Hello, everyone. Hopefully you can hear me. Yes. Okay. And give me a moment to bring up my presentation. Share screen. So you can see that, right? And okay. Hopefully everyone can hear me. I am Matthew Nielsen, a postdoc at Stockholm University, and today I'm going to be sharing with you research I've been doing with Carl Gotthard on how climate change is potentially shaping the evolution of phenotypic plasticity. But I'd like to start by reminding you all that seasonality is a really common phenomenon in many environments, and almost by definition, it means that organisms in these environments need to be able to adapt to a wide range of different conditions over the course of the year. And one of the major ways they do this is via seasonal phenotypic plasticity, where organisms will produce different phenotypes at different times of year, whether that is, say, deciduous trees losing their leaves in the winter, a wide variety of different sorts of seasonal color plasticity we see, or changes in many, many other traits. But one thing that unites the vast majority of these different seasonal plastic changes is that they need to be able to know what time of year it is and what kind of seasonal conditions they're going to be encountering to produce an appropriate change. And this can be a challenge because while for some species where plasticity is, say, rapid or easily reversible, they might be able to use the present weather to predict future weather, especially when thinking about long-term changes or irreversible changes, like I say, we're all probably familiar with the fact that current weather is very often a poor predictor of future weather, especially over long periods of time. So instead, many organisms, particularly at higher latitudes, use photoperiod as an indirect cue for seasonal changes. This works because photoperiod is a very reliable indicator of what time of year it is. And then that in turn, especially again at higher latitudes, can reliably predict what kind of weather organisms are likely to be encountering in the future. Now, at least historically, this has been the case, but unfortunately, thanks to anthropogenic climate change, that relationship is changing. Particularly as we're all familiar with, the world is getting warmer. You can perhaps see here how, in this case, I've graphed this or gotten maps of this for the Stockholm region of Sweden, one of the places I'll be working in this study, showing that it indeed has warmed over the past, several decades and is predicted to continue to warm into the future. And this warming is changing all sorts of different aspects of the abiotic and biotic environment. But one thing that climate change can't really affect is photoperiod. It's not changing how long the day is. And this can create a Q environment mismatch for organisms evolved under a particular range of temperatures that would be associated with a given photoperiod, but now there are different temperatures associated with that photoperiod, and they might be producing an incorrect phenotype because of that. So this leads into the big question of my talk, which is, can evolution correct for these climate change created Q environment mismatches we might be seeing in seasonal plasticity? So, but before I get to that question in detail, there's one more sort of additional feature of seasonal plasticity I need to introduce. And this is the fact that for most organisms, seasonal plasticity isn't a single decision the organism is making. This is really obvious for some cases, like say migratory birds, which when they migrate to their winter grounds, they then also have to choose when to migrate back from those locations. It's also something that we see in, like, say, for example, annual plants, where in addition to choosing when to flower and how long to flower, they also have to decide when they're going to germinate from seed. And all these different stages of their life cycle, they need to decide when to time those. And a final example of this, which incidentally is the species I'm going to be talking about today, comes from Parageozeria, the speckled wood butterfly. This is a European butterfly species. I've been studying it in Sweden, where depending on where it is in its range, it has between 1 or 2 generations each year. And one of the interesting features of this butterfly is that it can diapause, it can overwinter either as a larva or as a pupa. So there are 2 different life stages it can enter diapause in. And this reflects the fact that this species has really complicated seasonality. This sort of was teased apart by work that was done by Søren Nielsen and colleagues during the 1980s, and is summarized in this table for the southern Swedish population, where, as I had been mentioning, there is winter diapause in either larva or pupa. But in addition to that, they can also have plasticity of larval duration during the summer, where early in summer, they will develop quickly and directly in both life stages, but in the later summer, they'll actually slow down their larval development, which changes when they enter pupation. So they also are making these plastic decisions about seasonality in the summer as well. And if we look at the actual data that underlies that, there's a lot going on in this original graph, so don't worry too much about all the details. But what I want you to notice in this original data is specifically that they were able to manipulate photoperiod in the lab and recreate this sort of seasonal shift. So showing that photoperiod is really the key cue that they're using for these different forms of seasonal plasticity. So at long photoperiods indicating summer conditions, they develop directly and quickly as both larvae and pupae. And then as photoperiods get shorter, indicating progression through summer into fall, they start delaying their larval development. That's that sort of late summer change. And then they start entering diapause as either pupa or larva. Now, they also collected data from a central Swedish population. This is a population that only has 1 generation per year. But even in this population, they still have both this larval and pupal plasticity in development time. So even though the life cycle is very different in this population, they still show at least potential for these same sorts of plasticity. But that was all historical data collected during the 1980s. And now the situation is potentially somewhat different. So, and climate change is certainly happening here as well. So here's data I gathered from sort of available weather stations for the historical site, showing that for both populations at most times of year, the mean daily high temperature has increased between sort of the decade preceding these studies and the most recent decade. A little bit more in southern Sweden on average than it has in central Sweden, but both have experienced substantial climate change. And that's something means that this is a great study system for the kind of questions I want to be asking. So has the photoperiodic reaction norm for these 2 different sort of seasonal developmental plasticities evolved over the past 30 some years? And sort of as sub-questions of that, have these 2 different larval and pupal reaction norms evolved similarly to each other, and have these different populations evolved similarly to each other or not? Now, to get at this question, I initially went back to the original data that Søren Nielsen and his colleagues had collected for both these populations. And now they collected a lot of data. They raised a lot of caterpillars under a lot of different photoperiods, more than we could logistically do right now. So I specifically chose to focus on a narrow range of photoperiods that captured some of the most interesting phenomena in the historic data. So starting with photoperiods where we knew they would directly develop in both developmental stages, capturing that sort of late summer delayed, slowed larval development, and then catching also the start of pupal diapause. So for those photoperiods, we sought to recreate the original experiment using contemporary populations. So we collected adults from the same sites as had been used historically, and then raised their offspring in the lab under the same conditions that were used in the historical experiment. So the same temperature, which is 17 degrees Celsius, on the same diet. We were even able to use some of the same plastic cups that had been used in the original experiment. then we measured some of the same traits that they had. So both larval development time and pupal development time. And now for pupal development time specifically, we're interested in whether they're entering diapause. This is relatively bimodal. And so we specifically used the threshold of if they have been in pupa for more than 20 days, this means they've entered diapause. This has worked well in previous or subsequent studies of this species. And it conveniently means I can actually present results to you today because not all of the pupae have actually finished pupating yet. They're in the lab not too far away from me right now. But as basic hypotheses going into this research, we predicted that given climate change and the warming temperature, this butterfly should have access to a longer flight season than it did in the past. And this should result in a leftward shift of both reaction norms. So they start these sort of slower development and diapause later in the year than they would otherwise. That was sort of our initial hypothesis going into it. So we had some idea of what we might think to see. And so the first results I'm going to show you are for the larval reaction norm. So larval development time, and for the southern population first. So to walk you through this graph, on the x-axis, we have the photoperiod they were reared at. This was identical for both populations. The points are just slightly offset so you can see them more clearly. And then the y-axis is larval development time. The red circles indicate the historic data, and the blue triangles indicate present contemporary data. And then the larger solid symbols indicate the means for each treatment. And what we can see here when we look at the southern Swedish data is that for most photoperiods, these means are virtually identical. So there really hasn't been change under most conditions. However, there is a difference, and it's a pretty substantial difference at 17 degrees Celsius, or sorry, 17 hours of light per day, where they're now developing faster than they used to. And when we look at on average, and when we look at the actual data points, what it appears is going on is that in the historic population, you had a mixture of individuals that were directly developing as larvae and then entering this sort of late summer delayed, slowed larval development. But in the contemporary population, there are almost no individuals entering this sort of this delayed development. And so when we look at this overall, it seems like we are seeing some of that rightward or leftward shift that we expected. And they're now sort of entering this slower larval development later on in the year and at shorter photo periods. However, when we now look at the central Sweden data, we actually see a very different result. So this is a somewhat different part of the reaction norm we're capturing, but also something very different is happening. So this is structured the same way, but if we look at the historic data at the shortest photo periods, it's still pretty much the same. But at intermediate photo periods, they're now developing faster than they were historically. They're developing a little bit faster overall. And then at the longest photo period, this one where you had the mixture of the two, they actually seem to be developing a little bit more slowly in the contemporary population. So there's overall a bit faster development, but this also seems to reflect a rightward shift of the reaction norm rather than a leftward shift like we had predicted. So what we're seeing here is we are seeing potential evidence for evolution of the reaction norm over the last 30 years in this species, the reaction norm for larval development time, but it's actually happening in opposite directions in these two different populations we're studying. Now I'm going to show you the results for pupal diapause, which are a little bit simpler. So right now this is South Sweden data I'm showing you, and in dark, I'm showing you the proportion of pupa that entered either diapause or direct development pathways at the different day lengths we studied. And in the dark indicates direct development, light diapause development. And if you look at this, you can see the statistics aren't done, but these are really, really similar reaction norms. At long day lengths, they're always direct developing in both populations. At short day lengths, they're always entering diapause in both populations. And at 16 hours of light, there's a mixture in the historic data, and it's a really similar ratio in the contemporary data. So we're not seeing much change in the pupal reaction norm for diapause. And if you look at the central Swedish population, so this more northern population, it's a pretty similar result. In this case, there's maybe a little bit of difference. Particularly, we're seeing some direct development at a short photo period that we weren't seeing in the historic data. But other than that, overall, the data is quite similar. So we're not seeing any strong evidence for a shift in the reaction norm of the pupal diapause. And that's actually quite a difference what we're seeing in the larval development norm. So, and this difference is one of the key results I want to take away from this, is that We are seeing evidence for evolution potentially as a response to climate change over the last 30 years, but it's not occurring in all life stages and in all sort of the different plastic reaction norms. And one of the things this means is that often organisms might have multiple options for how they can evolve in response to climate change. And evolution of one reaction norm, in this case, life history, might actually serve to make the adjustment that's needed and mitigate the need for evolutionary change in another reaction norm, in this case, pupal diapause. I think the other big lesson to take away from these results is that we're seeing different evolutionary changes in different populations of the same species at different sites. This is an important message when thinking about climate change because it means we might be seeing very different evolutionary responses to climate change in different places. And this might be a more general pattern or something for future studies to look into. And for this particular study where we're seeing opposite directions of change in different populations, we're still working on explaining why that is, but some of that difference might lay in the difference in volatinism and number of generations per year in these populations. Where if you recall, I said in the South Swedish population, historically, they had 2 generations per year. And in the Northern Swedish population, they only had 1. So climate change might be acting on those differently. And it's also possible that even that volatinism itself is shifting. And we're going to be looking into data, trying to find data to see if we can test that in the wild, but where maybe there might actually be producing more generations now, at least in the southern population in some years. But that's the future research on that I hope you'll be able to see next time I talk about this research. For today, what I'd like to do is thank the people who helped me with this research, especially Sura Nuland, who gave us access to historic data for this research. I'd like to thank you for listening to my talk. I'd like to note that we did pre-register this study, and if you'd like to see more data about information about that, you can follow the indicated link. And then if you have any questions, I look forward to taking them. And if we don't get to them here, I'm happy to respond to questions about this via any of the indicated contact information.
Great talk. We already have some questions coming in, so we'll start with Carol Boggs, who asked, why do you think that some pupae are direct developing now at the shortest photo period?
So that's an excellent question. As I said, we're still working through statistics on there because the statistics we pre-registered, it turns out, don't actually work on our data for some reason. So it's a little bit of an open question exactly what's going on there. But my best guess is that if it's not just a fluke, there might actually be a little bit less sensitive to photo period than they were historically. There might be some dampening of that reaction norm because we're actually getting a little bit more diapause at longer photo periods. So that if I have to explain it, that would be the explanation I give right now.
Great. And follow up from Susanna Wajmar, which says, cool talk. I'm curious whether you think the rate of development or pupil diapause would be affected by the rate of daily change in day length as they might experience in the field.
Yeah, no, that's a very good point. These are all under constant conditions in the lab because that's what was done historically. But it definitely is the case that's where one of the reasons we don't actually look at pupil duration necessarily, because that is something where in the field they'll be in a very different condition by the end of it. And the same actually goes for larval diapause. So yeah, that's definitely a very important point. And that's actually one of our main ideas for the adaptive nature of this summer larval delay is it gets them to sort of a later, shorter photo period where they will enter pupil diapause correctly. And that might be even why adjusting this timing is enough, because now they're going to reach pupation at an appropriate time, even in the new climate. So that's a really good point.
And Samuel Shiner asks an interesting question that you focused on changes in mean temperature, but what about temperature variability, especially extremes in temperature?
So that's a really fair point. Temperature variation is another thing that's starting to get traction in climate change literature. One of the reasons for focusing on the mean is that we actually have much better documentation for changes in mean temperatures than we do climate variation. There's really clear records of that. We know that's been happening. Climate variation, while we're getting more extremes temperatures, part of that is just because we're shifting the means. If you shift the mean, you're going to get just more extreme, even under constant variation. So that's, I think, really the reason to focus on mean temperatures as the context here. That said, it's definitely worth looking into if there's been shifts in the variation of temperature over this time for the same populations, and we can hopefully report that data in the paper.
Great. And I don't see anything else that's come in. Oh, one from Julia Saltz. Great talk. Any thoughts about how changes in diapause voltism will alter future evolutionary trajectories, and any way to test for this?
That's a great question, because one of the things I didn't get at is that, of course, whether or not you should enter diapause really depends on how many generations you have per year. And their ideas regarding, like, if you change the number of generations per year, that can actually really, like, that's something that can potentially be entirely reversing the direction of selection on when you should enter diapause and how quickly you should develop. So that's something that I can't necessarily say what's going on in this system right now, but it's something I really hope to look into, because that could help explain why we have these really divergent change in reaction norm if there's sort of some incidental, at least, anecdotal evidence that maybe they are sometimes producing a third generation in the South, where in the central population, they might really be just sticking to producing one generation per year. So yeah, hopefully that addresses the question.
Yeah, I think that's great. I'm gonna stop it here.
Yeah, just a few seconds left before our next speaker. Thank you. So our next talk is a pre-recorded talk, I believe. I'm not sure if the speaker is here to answer questions afterwards or not. I guess we'll find out, but I'll introduce now Patrick Rohner, who's going to be talking to us about the developmental underpinnings of sexual dimorphism, sex-specific plasticity, and the resolution of sexual conflict in dung beetles.
Yeah, I'll play the video. If something's not working, just give me a shout because I cannot see.
Hello, everybody. Today I'm going to talk about the developmental underpinnings of sexual dimorphism, sex-specific plasticity, and the resolution of sexual conflict in dung beetles. Now, a central aim of evolutionary biology is, of course, to explain the vast diversity in organismal shapes, sizes, and lifestyles across the tree of life. One phenomenon that contributes greatly to this diversity is sexual dimorphism. Now, males and females most prominently differ in their reproductive investment, but this often also leads to divergences in other traits, such as overall body size or the development of secondary sexual traits. Sexual dimorphism is thought to evolve from a monomorphic ancestor by the action of either sexual selection or due to some form of sex-specific natural selection. However, sex-specific selection is not the only requirement for the evolution of dimorphism. This is because males and females often share almost their entire genetic material, and selection in one sex often leads to correlated responses in the other. If traits that benefits males cause fitness costs in females, this can cause interlocus sexual conflict. This means that in order for sexual dimorphism to evolve, such between-sex genetic correlations have to be overcome somehow. Now, because sexual dimorphism is such a common phenomenon, the resolution of genetic correlations has received a lot of attention. Conflict can, for instance, be resolved if genes with sexually antagonistic effects are genetically linked to sex determination loci or located on the sex chromosomes. Other mechanisms include the adjustment of sex ratio of offspring or the silencing of maternal or paternal alleles via genomic imprinting. And a further mechanism is, of course, to selectively express genes either in males or in females. However, sex-specific fitness optima usually depend on the environment an individual is exposed to. A mechanism that resolves conflict should therefore also account for environmental variation. In addition, conflict is not limited to sex, but can also arise when environmental conditions favor alternate phenotypes, as for instance in species with polyphenic development. Conflict can also arise within a single organism. All 6 legs of this male harlequin beetle, for instance, share a highly conserved and overlapping developmental program. This individual therefore must somehow find a way to only elongate its 4 legs while keeping the mid and hind legs short. So while there are several types of genetic correlations and conflict, the developmental mechanisms by which these are broken are still poorly understood. To better understand how sexes, morphs, and different serial homologs within an individual manage to differ in trait exaggeration, we here use Digitodontophagus gazella as a paradigm. Oops. This species is especially interesting to study for our purposes because it develops interesting secondary sexual traits. This first and foremost includes a pair of horns that is developed on the head of males, but maybe more importantly so, males also develop pretty long and elongated forelegs that they use during copulation. The elongation of these forelegs is particularly pronounced in large males. That is, tibia length shows a typical hyper-allometric relationship. Females, on the other hand, here indicated in blue, produce much shorter and stouter forelegs that also have a less steep scaling relationship. And we think that because females use their tibia for the construction of underground breeding tunnels, the broadening of the tibia may be an adaptation to a fossorial lifestyle. The hind tibia, on the other hand, is much shorter than both the male and the female tibia, which gives us access to 4 different, more or less exaggerated versions of the same homologous trait that is expressed in different contexts. We can use these contrasts to try to learn something about how traits vary in a sex, segment, and environment-specific manner. More specifically, we first asked, what are the transcriptomic underpinnings of tibia exaggeration? To do so, we wanted to compare the transcriptomic differences caused by nutritional plasticity, sexual dimorphism, and segment identity. This process starts with the cow because we needed dung to rear these beetles under laboratory conditions. And we then dissected tibial tissue from pre-pupae, which is the stage at which the future tibia is formed. We then extracted and sequenced all the mRNA in our samples and mapped the reads onto a de novo assembled transcriptome and tried to annotate all of the genes. Now, if exaggeration is driven by a shared set of core exaggeration genes, we expected the same genes to show differential expression in all of the 3 of these comparisons that we made. For instance, we expected genes related to insulin signaling or tibia development to be shared across all of these contrasts. Overall, we found around 1,200 genes that were differentially expressed across the comparisons that we were interested in. For simplicity, however, I will only show you the number of transcripts that had a higher level of gene expression in the tissue that develops into the larger structure. We found that the overlap between contrasts is pretty small, in that we only found 21 transcripts to be shared across all contrasts. That is only a scant 0.02% of all the differentially expressed transcripts that we found across the whole experiment. Furthermore, and unfortunately, these transcripts did not map to any known genes in either Ontophagus, Tribolium, or Drosophila, so their function remains unknown. They could, for instance, be taxon-restricted genes that only involve a specific lineage, but they could also be artifacts from our bioinformatic analyses, so their function really requires further scrutiny. One of the genes that showed up in 2 out of the 3 contrasts was double sex. This gene showed higher levels of expression in large compared to small males, and it had higher levels of expression in forelegs compared to hind legs. Previous studies further show that double sex is a highly conserved somatic sex determination gene that acts as a transcription factor. The interesting property of the gene is, however, that it undergoes sex-specific splicing. This allows the different sex-specific isoforms to target different target genes, therefore uncoupling male from female gene expression. In addition, double sex has also been demonstrated to play a major role in the regulation of sexual dimorphism and nutritional plasticity in other species. We were therefore interested in testing whether this gene contributes to the context-dependent development of tibia in our species that we were studying. To characterize double sex's function, we can now use RNA interference to knock down gene expression in all isoforms simultaneously. Knocking down double sex expression decreases tibia length in males, but increases it in females. This suggests that double sex mediates sexual dimorphism and that it plays a role in resolving intersexual genetic relations. We also found this to be the case for the length of hind legs, but to a lesser extent. This indicates that the function of double sex may not only depend on sex, but could also depend on the trait in question. As I mentioned earlier, the long forelegs in Gazella are pretty unusual for the kind of beetles that we regularly study. We therefore were interested in whether double sex may also contribute to species differentiation. We therefore repeated double sex RNAi with the close relative Ontophagus taurus, which does not develop such elongated forelegs. And we found that double sex also impacts foretibia size, introduces it in males and has almost no effect in females. And we also found that it did not really affect hind leg length at all. Together, this suggests that the downstream effects of double sex evolve in a trait, sex, and species-specific manner. However, size is only one aspect of exaggeration. In addition, we were very interested in shape. So we used a geometric morphometric approach to test if double sex also has effects on shape. On the left side, you see the result of a canonical variant analysis. This is an ordination technique that finds the axis that best differentiate between groups. Here in red, we see control males. In dark blue, we see control injected females. And in the middle, we see males and females that have been injected with double-stranded double-sex RNAi. When knocking down double sex, male tibiae become more broad and less elongated, while we find the opposite pattern in females. This shows us double sex also mediates sexual shape dimorphism and not just sexual size dimorphism. Knockdown effects were again stronger in forelegs compared to hind legs, indicating that again the function of double sex is not sex but also segment specific. So far we have shown that double sex mediates sex, trait, and species-specific exaggeration in size and shape of homologous structures. This contributes potentially to breaking down genetic correlations between sexes and serially repeated structures within a single individual. However, we were then interested in whether double sex could also play a role in phenotypic plasticity. In an additional experiment, we then reared animals on varying amounts of food and crossed this nutritional treatment with a double sex injection. These data show that double sex knockdown decreases allometric slopes in males, but increases the steepness of the scaling relationship in females. Double sex therefore not only mediates sexual dimorphism, but does so via manipulation of sex-specific plasticity. This is also evident when we look at shape. Here you see two plots that show a shape projection of tibia and horn shape onto the shape vector defined by sex differences. This is a little bit complicated, but to put simple, individuals with a high score on the y-axis have a more male-like morphology, while individuals with a low score have a more female-like morphology. First of all, we see that in animals injected with buffer solution only, sexual shape dimorphism increases with nutritional plasticity. This was especially pronounced in horns. It's mostly driven because horn expression is limited to males that only have access to high nutrition. Secondly, we see that the effect of knockdown of double sex also increases with nutritional plasticity. This suggests that double sex affects sex-specific plasticity in tibia shape. To further test this, we can now compare the direction of shape changes related to nutrition responsiveness shape between males and females. In buffer-injected individuals, the correlation between male and female, which are here shown in black, is pretty low. This is not surprising because we know that males and females differ in the development of secondary sexual structures and that the scaling of those structures also is different between the sexes. However, when we knock down double sex, these differences appear almost completely. This suggests that double sex is necessary for the sexes to follow independent nutritional responses. Now let's conclude. Using RNA sequencing, we found that trait exaggeration in homologous structures in different contexts is not necessarily mediated by an overlapping transcriptomic landscape. However, using functional genetics and geometric morphometrics, we found that double sex underpins sexual dimorphism in size and shape of all structures investigated. Double sex and its sex-specific isoforms therefore seem to be utilized to regulate optimal trait expression depending on the sex, location, species, and environment the trait is expressed in. This suggests that double sex contributes to the resolution of genetic correlations in several contexts. Because double sex is found in all hexapods, its function evolves fast, and alternative splicing seems common, double sex is therefore likely to be a major contributor to phenotypic variation in insects. And lastly, I think these data show that studying developmental underpinnings of adaptive trait differentiation can really further our understanding of the evolution and diversification of sexual dimorphism more generally. And with this, all that is left for me to do is to thank you for your attention, the organizers for putting together this conference, and of course the Mojek group for providing a very stimulating environment despite an ongoing pandemic. Thank you very much.
That worked okay?
Patrick is here for questions if people want to throw those in the chat or the Slack. Okay, first question. Is there any evidence of sex load or unresolved sexual antagonism in the beetles?
So we only started at really looking at the causes and consequences for sexual dimorphism in these species. And morphologically, I do see, or we do have some evidence that fitness optima probably differ between sexes, which is based on their behavior. But there's probably also a form of behavioral conflict because males use their long forelegs to hold on to the female during copulation. And just by looking at it, it looks as if males would coerce females to mate with them. But we are only just in the beginning of investigating that. So it's a good question. Thank you.
And we have another few. So the next is, do you think that nutrition affects DSX activity due to the lack of amino acid in poor diet, or is this a direct regulation through nutrient pathways?
We don't know. Now, of course, from the condition dependence hypothesis, or from that background, we would, of course, expect this to be the case. However, in this specific instance, we just don't know yet. Double sex has been, and its regulation has been heavily studied in Drosophila and a few other species. But how double sex expression is linked to nutrition or other environmental conditions really is poorly understood so far. There are only a few studies that document it at all. So I think that's an exciting way to look into this. But yeah, so unfortunately, so far, we just don't know.
Kim Hoke says, fantastic talk. To what extent do you think double sex has independent effects on different traits? Is it easy to decouple effects on different body parts?
So I now showed you only some results that we have for horns and for legs, but we measured a bunch of different traits too. And it appears that in every single trait that we look at, that includes the thorax, other legs. Also, I did also some measurements on wings, including overall body size, development time, these kinds of things. And double sex seems to have an effect on sexual dimorphism in all of those traits. So it seems that double sex really is a major driver of sexual dimorphism and that this evolves really fast. Now we have only knocked down double sex in a few species and we don't have that many comparisons between species, but it seems to evolve really quickly.
That's great. And in answering that, you actually answered Ava Fisher's next question, which is double sex important for the development of sexual dimorphism in general? And how does this inform our interpretation of these differences? If these effects are really far upstream, could they be affecting nearly all sex differences? Which it sounds like maybe it is.
Yeah, that's probably what it is. So what eventually then evolves probably is how double sex itself changes in expression between males and females, between different structures, and depending on the environmental context. But what we don't know yet is whether the way tissues react to double sex levels, whether that's the case too, that we don't know yet. But I think it's an exciting topic to look at in the future. But yeah, it seems to be a really far upstream and seems to be quite basic.
That's great. Yeah, I was wondering if you had any clues as to what during development is mediating these context-specific and sex-specific differences.
We don't know. We don't know. We also injected double-stranded RNA towards the beginning of the third instar, which is the last instar. But these beetles have been, how old would they be? They may be 10 days old, and total development lasts like, I don't know, maybe 20. So it's not something that starts at the very beginning. So yeah, but we really don't know when exactly these effects kick in, and whether that's also something that differs between traits or species. Yeah, it's a good question.
Very cool. I'll stop us there to keep us on time.
Yeah, I think that's perfect. Thank you, everyone. So our next speaker is Libby Davenport, who's going to be talking to us about whether or not spontaneous mutations in Daphnia pulex have affect fitness equally at two different temperatures. So I'll turn it over to Libby.
Thank you. Hi, everyone. My name is Libby Davenport. I'm a first-year PhD student at the University of Michigan. Today, I will be presenting my research on the fitness effects of spontaneous mutations, which I completed at the University of South Carolina in the lab of Dr. Jeff Dudica. My main research question is, do spontaneous mutations affect fitness-related traits equally at two temperatures? In general, spontaneous mutations fuel evolutionary processes by introducing new genetic variation to populations. And these mutations can arise due to replication errors, and their effects are typically neutral or deleterious, but can occasionally be beneficial. And in some cases, there's even evidence of condition dependence for mutations. These condition-dependent effects mean that the magnitude of these effects of mutations depend on the environment and abiotic factors such as temperature. It's also worth noting that temperature can affect protein stability and function. So we predicted that elevated temperature would disrupt protein folding, leading to diminished stability in a protein that ultimately is less able to tolerate the effects of an amino acid substitution. So we predicted that elevated temperature would exacerbate the deleteriousness of spontaneous mutations. And with respect to fitness-related traits, such as the intrinsic rate of increase, we expected to see reduced fitness due to spontaneous mutations that have accumulated over the course of numerous generations, as well as to observe a more substantial fitness reduction at a higher temperature. In order to test this hypothesis, we studied the freshwater microcrustacean Daphnia pulex. Most Daphnia reproduce via cyclical parthenogenesis, which involves switching between modes of sexual and asexual reproduction. However, some clones of Daphnia pulex are actually obligately asexual. So these clones are still able to produce resting eggs like other Daphnia clones, but these resting eggs, or ephipia, are diploid rather than haploid. So this is highly advantageous for experiments in the lab involving spontaneous mutations, because these resting eggs can be preserved at -80 degrees Celsius and later hatched. So we are effectively saving genetically identical ancestors, which can be ultimately compared to the current generation of different genetic lines or clones of Daphnia. So Daphnia pulex, particularly these obligately asexual clones, can be used for mutation accumulation experiments. So mutation accumulation experiments have been used for decades to study phenotypic and fitness effects of spontaneous mutations. And this has been done with a wide range of model organisms, including Daphnia, Drosophila, Arabidopsis, and others. These experiments begin with the establishment of different lines from a common ancestor. For Daphnia pulex, this involves beginning with an original female clonal ancestor. So you can take the original offspring from a female and establish different mutation accumulation lines. These mutation accumulation lines can be maintained in the lab under standard laboratory conditions at 20 degrees Celsius. So all of these different lines vary depending on their genetic differences due to spontaneous mutations that are occurring and accumulating over the course of all of those generations of asexual reproduction. Several years ago, the Dudica lab established mutation accumulation lines for the clone Lis. At the time of the establishment of the mutation accumulation lines, ancestral resting eggs were also preserved and saved for later. So these resting eggs can be hatched at a later time period. So I wanted to utilize these resting eggs of the clonal ancestor in order to compare a later generation, which happened to be generation 37, to determine the effects of spontaneous mutations. So I hatched the clonal ancestors and then used the 37th generation for a phenotypic assay. And since I'm interested in the effects of temperature as well as spontaneous mutations, and I wanted to test for an interaction between those two factors, I did the phenotypic assay at 2 temperatures. 20 degrees Celsius, because this corresponds to the conditions for the mutation accumulation experiment itself, as well as 24 degrees Celsius, which aligns with climate change projections. And it's worth noting that with regards to our original hypothesis, we again expected to see reduced fitness in these mutation accumulation lines compared to this clonal ancestor, and that we planned to observe a more substantial fitness reduction at a higher temperature, so at this 24-degree treatment. Now, prior to the phenotypic assay, I hatched the ancestral ephipia of the Lis clonal ancestors and established 12 control lines. Once I had hatched the controls, I obtained the 37th generation of 21 mutation accumulation lines. And I raised all of these lines under standard laboratory conditions for 3 generations to standardize for maternal effects. This included containing 1 individual per beaker, as well as daily quantitative feeding, and transfers to clean beakers every other day. Once I had an adequate number of individuals, I set up the first block of the phenotypic assay. This was a temporal block, and it consisted of 2 temperature treatments, again, 1 20-degree treatment and 1 24-degree treatment. For each temperature treatment, there were 10 individuals per line, and this was for the 21 mutation accumulation lines and 12 control lines. After block 1, we shortly completed the temporal block 2 following the same protocol. During the phenotypic assay itself, so during block 1 and block 2, we tracked survivorship and reproduction over the course of the first 4 clutches for each individual. So we collected survivorship and reproduction data on over 1,200 individuals. This involved counting over 81,000 neonates in total for block 1 and block 2. So with the survivorship and reproduction data, we were then able to estimate little r, so the intrinsic growth rate. And this was our index of fitness. And we did this using a Newton-Raphson approximation. In addition to these fitness-related traits, we also considered morphological traits like body length at maturation. So for the first temporal block, we measured the body length at maturation for all of the individuals. Now, moving on to the results. We found some very interesting trends related to spontaneous mutations and temperature for reproduction for the control lines and mutation accumulation lines. So here we have the number of offspring in a clutch plotted against the day of clutch production. And this is split between the two temperature treatments. So 20 degrees on the top panel, 24 degrees on the bottom panel. Each point represents the average for a line. Mutation accumulation lines are in green, control lines are in black. For the first clutch, shown here, there was no significant difference between mutation accumulation lines and control lines with respect to clutch size. However, for the second clutch, there was a significant difference for clutch size, with control lines producing significantly more offspring than mutation accumulation lines. This was also observed for the third clutch and the fourth clutch. These trends in clutch size were observed at both temperatures. However, at 24 degrees Celsius, we observed that reproduction was accelerated. So the interclutch intervals were much shorter, and all 4 clutches were produced earlier in comparison to the 20-degree treatment. We also found it very interesting that mutation accumulation appeared to increase variance among lines, and this was most evident for the third and fourth clutch. Over the course of these 4 clutches, the control lines produced more offspring at both 20 degrees and 24 degrees. So spontaneous mutations had a negative effect on the total number of offspring produced. Temperature, meanwhile, also had an effect on the total number of offspring produced over the course of those 4 clutches. So at the warmer temperature, fewer offspring were produced for both control lines and mutation accumulation lines. But with respect to our original hypothesis, there was no significant interaction between temperature and mutational history. We saw similar trends for body length at maturation. So control lines generally had individuals that were larger on average than the mutation accumulation lines. And this was true, again, at 20 degrees and 24 degrees, with no significant interaction between temperature and mutational history. So if we move on to the survivorship data, we notice that both mutation accumulation lines and control lines had high survivorship at both 20 degrees and 24 degrees. So our estimates for little r were primarily based on the reproduction data. So those estimates of r, which were used as an index for fitness, are represented here in this graph, plotted against generation, with our ancestral lines, or those control lines, at generation 0, and then our mutation accumulation lines with an average generation number of 37. We can clearly see that there's an effect of temperature here. So all of the points in red represent the average fitness of the lines at 24 degrees Celsius, so fitness increased at this warmer temperature. The horizontal lines here only represent the mean of the control lines. We also observed a similar trend regarding the variance. So once again, spontaneous mutations increase the variance among lines. And this variance was greater at the warmer temperature. But in order to test our hypothesis regarding the interaction between temperature and mutational history, we rescaled fitness to relative fitness. And with this regression, we're able to see that the effects of spontaneous mutations were roughly the same at both temperatures. So the magnitude of those fitness differences between ancestral and mutation accumulation lines was approximately the same at 20 degrees and 24 degrees. So this does not support our original hypothesis, but it does answer our central question. Moreover, we found it particularly interesting that some mutation accumulation lines actually had a higher fitness than the control lines. So if we highlight those here on a reaction norm, we notice that although the majority of mutational accumulation lines had a reduction in fitness, so here, the solid line represents the mean of the controls, and all of the gray dashed lines represent the individual mutation accumulation lines. More mutation accumulation lines fall below the average of the controls. However, again, we found some trends that suggest some condition dependence in several mutation accumulation lines. Also, several mutation accumulations had higher fitness at both 20 degrees and 24 degrees than the ancestors. And this is unusual for mutation accumulation experiments. It's possible that we are seeing these beneficial effects because we conducted the phenotypic assay at a relatively early generation number. So once again, we completed the phenotypic assay at roughly generation 37. Other studies have waited until generation 100 or even 200 to conduct a phenotypic assay to determine the fitness effects of spontaneous mutations. So it's possible that by conducting the phenotypic assay a little bit earlier, we're still seeing those beneficial effects before they are overwhelmed or overshadowed by more deleterious mutations and more deleterious effects. And so the key takeaways from our study was that there was no evidence of a significant interaction between temperature and mutational history with respect to fitness-related traits. So an increase in temperature did not result in an increase in the deleterious effects of spontaneous mutations. However, there were main effects of elevated temperature, as well as main effects of spontaneous mutations. So we observed a decrease in the body length at maturation at the warmer temperature, as well as a decrease in the body length at maturation for mutation accumulation lines. We saw earlier production of the first 4 clutches at the warmer temperature, as well as a decrease in clutch size at the warmer temperature, which then influenced the total number of offspring produced at this warmer temperature. So there was a reduction in the total number of offspring at this warmer temperature. Spontaneous mutations also caused a decrease in the total number of offspring produced. However, the magnitude of this difference between control lines and mutation accumulation lines stayed about the same at both temperatures. And with respect to our estimates of little r, temperature increased these estimates, whereas spontaneous mutations ultimately decreased our metric of fitness. So overall, the magnitude of the trait differences between ancestral lines and mutation accumulation lines was approximately the same at both temperatures for clutch size, reproductive timing, body length at maturation, and fitness as estimated by little r. I would like to thank my undergraduate research mentor, Dr. Jeff Dudica, my senior thesis second reader, Dr. Carrie Wessinger, and all of the members of the Dudica Lab. And thank you all for attending my talk, and I'm happy to answer any questions at this time.
That was an excellent talk. All right, I'm just going to wait for questions to pile into either the chat or the Slack. Okay, first one's from Kelsey Lieberger. Do you think the earlier clutches didn't show differences between MA and control because the smaller number of eggs, or because the effects of the mutations don't take effect until later in life?
Yeah, that's a great question. So the first clutch generally is a lot smaller than the other clutches. So it's possible that we're just not seeing quite as much variation for the first clutch, and that potentially those effects are becoming much more apparent with those later clutches, since there's a greater clutch size, so there can be more variation among those. One thing that I didn't mention was that there was a difference in the timing of first clutch production. So with mutation accumulation lines producing clutches later than the ancestral lines.
It's really cool. Samuel Shiner asks, you may not have enough lines, but did you see any differences in the variability among temperatures?
Yeah, that's also a great question. So one thing that we could go back and do, actually related to the fitness estimates at the 2 different temperatures. So I showed you the reaction norm. One possibility that we could do is to bootstrap and get some confidence intervals for the reaction norm, and then better determine whether or not we're actually seeing a lot more variability among those different lines. So again, we could continue like looking into more of the variability at the different temperatures too.
Probably have time for one more question if someone wants to type it in the chat or the Slack. From Becky Fuller, the rate at which beneficial mutations come in seems somewhat surprising. I think of beneficial mutations as being pretty rare. Are there other possibilities in there as to what might be going on with those higher fitness? Adaptation to lab environment is posed as a possibility.
Yeah, so since we completed this at generation 37, this still allows some time for beneficial mutations to arise, but I definitely see your point that that is relatively a rapid occurrence of beneficial mutations. So it would be interesting to test or conduct a phenotypic assay at a later generation number and compare it to these earlier results. I think that would give us a better indication as to whether there is any sort of adaptation to these lab conditions. But based on these results, it's kind of hard to say for sure how those beneficial mutations arose.
That's great. I'm going to hold all other questions to keep us on time, but there is another one in the chat if you want to get at that one, Libby.
Thanks.
Okay, thank you. So our next speaker is Gonçalo Ferraz, whose talk has the title, Does the Damuth Rule Apply at the Local Scale? An Investigation with Tropical Forest Understory Birds. So I'll turn it over to you, Gonçalo. Can we hear? You're muted. Gonçalo, you're muted.
Okay, can you hear me now?
That's good.
All right.
Go ahead.
That's sharing. Let me see if this works. I am very sorry. All right. Okay. Good afternoon. My name is Gonçalo Ferraz. I am a professor of population biology at the Federal University of Rio Grande do Sul in southern Brazil. I would like to share with you the results of an investigation about whether a local sample of bird species conforms to an allometric relationship known as Damuth's rule. This work is the product of Yara Coriolano's master's research and is part of a paper that we would like to submit as soon as possible. Here is Yara with a motmot, which is a species not included in the sample for this study. Allometric relationships are recurrent, and there's many relationships that relate some trait with body mass with exponents of plus or minus 3/4. The recurrence of these 3/4 exponents is relatable to the dimensionality of energy acquisition and expenditure. One particular allometric relationship that matters to us in this work is Damuth's rule, which relates the population density to body mass with an exponent of minus 3/4. This means on the linear scale that as density goes down with increasing body mass, but it goes down less per unit mass as mass increases. On a log-log scale, this would mean that population density decreases with log population density decreases with log body mass with a slope of minus 0.075. This rule, this relationship has interesting theoretical implications for energy equivalence, resource monopolization, and more. If we combine the Damuth rule with Kleber's rule that says that animals increase their basal metabolic rate with body mass with an exponent of 3/4, the two rules combined lead to the notion of energy equivalence, that the amount of energy used per unit area should be invariant with body size. If, on the other hand, the power of the density increase is not exactly according to Damuth rule, but it has a higher exponent, less negative or even positive exponent, we should expect larger species to monopolize resources on a per area basis, backing Cope's rule. There's also the possibility that the combination of Damuth's rule with Taylor's power relationship between variance and mean of population size would predict a population variance mass allometry. So there is no doubt that this has interesting theoretical implications, but the empirical support for the Damuth rule with field data is puzzling. There's a wide range of exponents reported from less than -4 to approximately 1. And these findings change substantially, the amount of the magnitude of the exponent changes substantially because of a variety of reasons that include subsampling of small-bodied species in study compilations, different spatial scales of investigation, whether studies control or do not control for phylogenetic structure in the samples, and details about how the population density is estimated. Does the rule apply at the local scale? We ask this question because there's at least 2 major reviews that emphasize that there are shallower log-log slopes on local than on global investigations. So there's an expectation that this should be more difficult to find in local samples. We don't know whether this is because of local mechanisms that impede the relationship or just because statistically small sample sizes make the relationship more difficult to detect. On the right, there are 2 examples of a global relationship on top, and on the bottom, a shallower relationship between body mass and local population density from a sample of bird species in the Peruvian Amazon. So we would like to find out what happens at the local scale with a set of 44 species in an Amazon forest understory set of species from an upland site near Manaus, north of Manaus, in the center of the Brazilian Amazon. We did this investigation based on 5 years of field work with mixed net captures of birds in 2 areas, leading to about 8,000 species captures from 130 species and 31 families. In this analysis, we filtered the species to have a minimum number of captures and captures in all the years of our sampling period. And we're working with 6,844 captures of almost 5,000 individuals from 44 species in 13 families. We took a number of methodological precautions to work with this sample with the intent of avoiding bias in inference of the allometric relationship. We started out by estimating population density with a spatial capture-recapture approach, where we account for imperfect detection and home range size. We used the comparative analysis to recognize that species are not independent points in the analysis, accounting for phylogenetic structure in the data. And we incorporated intraspecific variability and our measurement error in both body mass and population density when trying to infer the allometric relationship. What I mean by estimating population density with spatial capture-recapture is that we worked with a method that is somewhat like distance sampling of individually marked animals with spatially indexed sightings. This approach estimates a number of individuals per unit area and home range size. So we're not saying that we have X or Y number of individuals in an unspecified area. We're actually estimating a density, a number of individuals per unit area. We did this for this study using the Secker-R package to fit models in a likelihood framework, and this package was developed by Murray Efford. 75% of the species from our set have between 2 and 20 individuals per square kilometer. The minimum is 0.5 individuals per square kilometer, and the maximum is 100. It became clear from the analysis that the species which have the highest number of captures are not the species that have the highest estimated density. To employ the comparative method, we obtained 1,000 different but equiprobable trees from birth tree. The illustration on the left illustrates one of these 1,000 trees. And then we looked for the relationship between body mass and the estimated population density. First, as a type of a null approach, we used an ordinary least squares analysis. And then we used the PGLS with the Brownian motion of trait evolution to investigate, to account for the phylogeny. And subsequently, we did a variant of the PGLS, which we'll call the PGLS Ives, that incorporates measurement error. All the analyses were done with the same 1,000 trees, and we did this using our packages Stats, Ape, NLME, and Phi tools. Our phylogenetic signal differed between traits. When we looked at values of the Bloomberg's K, it appeared that there was stronger evidence of phylogenetic signal for population density than for body mass, and we were surprised with this result. We didn't expect body mass to show more evidence of phylogenetic signal. And this is what we found when we did the three analysis. On the left, there is a graph that shows body mass on a log scale and population density on the log scale 2 on the y-axis. The dots indicate different groups of species. The black circles are antbirds, the most abundant family in the data set. The filled squares are ovenbirds and wood creepers, and the empty circles are other families with fewer than 7 species each. So the central results of this analysis were that the ordinary least squares analysis that accounts for observation error in the estimation of population density, but ignores phylogeny and intraspecific variation, indeed estimated a shallower slope, so a relationship between body mass and population density that doesn't conform very well with Damuth's rule. But as we included the phylogenetic structure and subsequently the measurement error in the analysis, we got closer and closer to the slope predicted by Damuth's rule. What does this mean? Because even though the slopes are close to what we expected, there is a lot of scatter around the regression lines. So there is substantial variation that is not explained by the regression model. However, when we did 1,000 likelihood ratio tests between the fitted PGLS Ives model, the most sophisticated model that we did, to compare it with a null PGLS Ives that has a slope fixed to 0, this resulted in a mean p-value of 0.054, meaning that we have almost a slope that is significantly different from 0 at the 0.05 level, for whatever that may mean. So we conclude that even with careful estimates of abundance, an ordinary least squares suggests a slope shallower than Damuth's rule, but including phylogeny brings us closer with the standard PGLS, and including measurement errors with the PGLS Ives analysis brings us even closer and with more precision in the slope or exponent estimation. So we come to the conclusion with this that our local data set seems to be compatible with Damuth's rule, and it does, at least in this case, apply at the local scale. I'd like to acknowledge Carol Schu, Gilberto Arellano, Luisa Figueira, Pedro Martins, and many others who, in the banding crews. Rena Maestri, Leandro Duarte, and Gabriel Nakamura helped with comparative analysis. Mark Carey and Igor Coelho were of great help with the abundance estimation. And I'd like to thank the ASN Asilomar 2021 conference organizers and all the colleagues who are attending. Thank you very much. Here are our contacts. Could you hear me well in the end?
Yeah, it was excellent.
Thank you.
Excellent talk. I'll just wait for questions to come into the chat or the Slack.
Mm-hmm. Okay.
I'll ask one while we wait. I actually wasn't incredibly familiar with this particular rule. And are there any clades or scales at which it's regularly broken?
I wouldn't say regularly broken, but larger samples. It has been initially documented with a large sample of mammals. And it has been reported to not apply so well to birds, in part, arguably, because birds move more and would use space in 3 dimensions rather than 2. But the more I read about this, the more it seems that the non-application with earlier bird samples may have been due to difficulty in parameter estimation, which is, it's sort of puzzling because the 1981 Damuth paper was not exceedingly sophisticated in estimating of parameters, and it did get the slope of minus 0.75, but with a huge sample size and with a lot of variation, with 4 orders of magnitude of variation in population density for any given body mass. So it's not a nice accumulation of points along the line.
Yeah.
There is if I may add, there is one point in our work that is not totally clear for us yet, which is this idea that there seems to be a stronger phylogenetic signal in the density values than in the body mass. That is surprising, and we're not too clear about what does that mean exactly, or whether it's something that we should capitalize too much on. I don't know if anyone has heard or seen attempts to look for a phylogenetic signal in abundance metrics, but I would be interested to hear anything about that.
Yeah, it's definitely surprising. I'm also surprised by that.
Mm-hmm.
I'll just note that we'll start the next talk on schedule in case people are moving around. So people might have if anyone needs to run to the washroom or grab a drink, you have a couple minutes to do it perhaps, or any other questions.
Mm-hmm.
There's one in the chat.
Yeah, density could be related to reproductive strategies, clutch sizes, for example.
Yes, it could. But as far as I know, no one managed to find enough nests of these species in this area to have interesting information about clutch size. João, would you suggest any source? Oh, I see the point. You're saying that because clutch size is conserved, density could be conserved in association with clutch size. That's an interesting point. Yeah, it deserves attention. Oh, I'll just turn on the slide with the contacts. And be ready to answer any questions. I think I got scared with a mistake in the beginning and then ran too fast, afraid of not having enough time. I'll just say to conclude that I find this topic particularly fascinating because when these relations recur a lot, they seem to impose limits to evolution, and that's why I find it so relevant to try to dig into even a local data set and try to estimate parameters as carefully as possible to see how recurrent these things really are.
Okay. Well, thank you, Gonçalo.
Thank you,
So moving on from there, our next speaker is Moira Robinson with the talk titled Domestication is Altered Within Plant Trait Variability and Diversity in Alfalfa. So I'll turn it over to, oh, sorry, it's Moria. Did I say that wrong? I read too quickly.
Moria. It's okay. It's not phonetically spelled.
Oh,
I respond
sorry.
to them all.
My apologies. Turn it over to you.
Thank you. All right, can you see the full slides? Great, let me get my pointer.
Yeah, it looks good.
Great, thank you. All right, wonderful. Well, thank you all for being here. I'm really excited to present some of the work I've been doing from my postdoc with Will Wetzel at Michigan State University. So to start, I'd like to thank the many folks who made this work possible. So in particular, funding through the USDA NIFA postdoctoral fellowship program and Michigan State University, and my advisor, Will Wetzel, and his lab at MSU. And also Tony Schillmiller, who was an integral part of many of the chemical analyses you'll see today, and the many undergrad research assistants who helped process samples and collect data for this work. And thank you to the Society and the organizers of this meeting. So humans are a powerful evolutionary force on the plants we grow. We've been shaping the phenotypes of plants for over 12,000 years. And understanding how plant traits have changed with domestication selection is really important, not only for human health, but also to understand the often very different ecology of crop systems. To date, most studies have focused on change in whole plant traits or average traits among repeated parts. For example, we know that plant size often increases with domestication, growth rate may increase, and nutritive or defensive characteristics of individual leaves, when we average them across those leaves, are often different between wild plants and the domestic cultivars we grow. And these scales and traits really dominate our current understanding of domestication selection in crop systems. But since the 1950s, there's been increased recognition that plants contain very high levels of trait variability among their repeated parts, and that this trait itself can be a heritable and ecologically and evolutionarily relevant component of the plant phenotype. So if we think, for example, about the different leaves within a plant, we can imagine that those leaves will be different for a number of reasons. They may be at different ontogenetic stages. They may have different levels of phenotypic noise or stochasticity in gene expression. They may have different levels of plasticity. And together, all of these processes scale up to create plant individuals that contain multitudes of trait variation. And this variability can affect plant resource acquisition and can affect the way that biotic interactors interface with these plants. So we know from studies of foraging ecology across scales that variability in important traits can be as or even more important than trait means to consumers. So adding this trait to our picture of domestication selection is really of fundamental and applied importance. Domestication is a natural evolutionary experiment, or unnatural, depending on your point of view. And it helps us understand how selection, perhaps on a subset of traits, can change an integrated organismal phenotype. So we can ask, how does trait variability fit in with these other traits that we know have shifted to form the domestication syndrome? And from an applied perspective, we know that low trait variation is a common feature of domestication and of crop systems. We grow single plant species, often with very depauperate genetic diversity among individuals, and this is often blamed for poor agroecosystem function and high levels of damage by herbivores. But we don't really know how domestication has affected trait variability at this within plant scale. And this is despite the fact that this scale, the spatial scale, can be really critical to many biotic interactions. So our big question is, has domestication selection altered levels of trait variability and diversity within plant individuals? So what might we predict? Well, our first thought was, we know agriculture values homogeneity and has reduced trait variation at other scales. So we perhaps naively hypothesized that this could also be occurring within plant individuals. We also know that mean and variance have a fundamental relationship, and Gonçalo mentioned this in his talk as well. So, and I'll talk about this a little bit more later, but selection for or against certain trait means could therefore increase or decrease the associated variability of those traits. We also know that inbreeding can increase errors in tissue development, so this could lead to greater trait variability among tissues of plants with domestication. So this is all to say, this is really an open question, but we can use what we know about domestication selection to predict very different outcomes for variability at the within plant scale. So the three questions I'll be discussing today are, first, kind of documenting this pattern: do levels of within plant among leaf trait variability shift with domestication? What can the nature of trait mean variance relationships tell us about these shifts and about the nature of domestication selection itself? And are differences in trait variability explained by leaf ontogeny? So the system we used to explore these questions was alfalfa. So alfalfa is a key forage crop with an 8,000-year domestication history. It's grown all across the world. And we selected 30 wild populations across the native range, up here on the map, and stratified across the 3 wild subspecies from which we have derived all of our modern cultivars. And then we also selected 30 domestic cultivars with a range of domestication histories. We grew 30 plants from each of 30 wild populations and 30 domestic cultivars. We were really going for breadth in trait variability across genotypes here. We grew them at Kellogg Biological Station in Michigan and sampled 9 leaves from each plant individual across 3 different ontogenetic stages, 3 different nodal positions on lateral branches. And on these 540 leaves, we measured a bunch of traits. So we looked at specific leaf area, leaf water content, trichome density, carbon content, nitrogen content, and their ratio, and the concentrations of 86 saponin compounds. And these are compounds of known defensive function in alfalfa. So for these traits, we measured the mean and also the standard deviation as a measure of variability. We also quantified 3 metrics of chemical diversity within these plants. So using these same 86 compounds, we looked at the alpha diversity of compounds within individual leaves, the gamma diversity, which is the pooled diversity across all leaves within the plant, and the dissimilarity in chemical composition from leaf to leaf, which is the beta diversity. So the goal here was really to gain a holistic view of the trait variation contained within these plant individuals and to ask whether levels of trait variability differ between domestic plants and their wild progenitors. So if we jump into this first question, what I'll be showing you here, because these different traits are measured on very different scales, I'll be showing you the effect sizes or the percent change in variability between wild and domestic plants on the y-axis. So above the 0 line means domestic plants have greater variability than the wild plants, and below the 0 is the reverse. And there are two different y-axis ranges here, but I'll put the effect sizes to discuss up top. So when we looked at these data, the first thing we saw was that yes, we did see differences in the magnitude of trait variability among leaves between domestic plants and wild relatives. So for example, if we look at specific leaf area, it's 70% more variable among leaves of domestic plants compared to wild relatives. We look at the concentrations of different saponin compounds, both individual saponin compounds and their total, and they are much more variable among leaves as well. We also see an increase in compound richness in domestic plants compared to their wild relatives. And we also see some declines. So we see that Simpson's diversity of these compounds is lower in domestic plants compared to wild relatives. And we also see that this dissimilarity in composition among leaves, or chemical beta diversity, is also about 16% lower in domestic plants compared to their wild relatives. So in sum, for this first question, we find that yes, domestication has changed trait variability at the within plant scale. We find that domestic plants have more variable leaf mass per unit area. That's the SLA result. We find that they produce more unique compounds at more variable concentrations. And from leaf to leaf, they have more homogeneous chemical compositions. So we were really excited about these results. And the next question we wanted to ask was to explore one of the potential causes for these shifts, and in particular, how intimately these shifts were tied to shifts in trait means and overall compound production. So just a little bit of background here. We know that trait magnitude and trait variability are related. So we know that a fundamental statistical law is that the mean and the variability often exhibit a direct relationship. This is Taylor's law. And we also know from chemical diversity literature that the total amount of secondary metabolites that are produced by a plant is often related to the total number of unique compounds within that mixture. So we wanted to ask, knowing that these two components of the phenotype are often linked, and given that we see these overall shifts in variability, do we have evidence that these are indirect products of shift in overall trait production, or are they an independent feature of the domestic phenotype? So just a few scenarios. If these two properties are directly related, then selection on means should bring along differences in variability. That's pretty intuitive. If the mean variance relationship has a different shape or slope between wild and domestic plants, then this could mask differences in trait variability. And if they're uncorrelated, then trait means and variability can change independently over evolutionary time. So our first step to looking at this question was to plot the correlations between mean and variability for our traits. So here are just a few, and without going into too much detail, this is simply to illustrate that mean and variability have complex relationships across traits. So they can be directly related, either positively, as for nitrogen and for chemical diversity, or negatively related, as for leaf water content. We also saw that they can have different slopes between wild and domestic plants, as you see here for trichome density, or the mean and variance might be uncorrelated for certain traits like carbon content. So to understand how these relationships might explain the shifts in variability we see or not, I'll show you the same results, but this is with trait mean or total compound concentration as a covariate in the model. So we would expect that if variability is being indirectly shaped by shifts in trait mean with domestication, then these effect sizes will go away. But if we still see an effect of domestication, then these shifts in variability are at least in some degree occurring independently of changes in trait means or overall compound production. So what do we see? Well, we see here that we retain certain shifts and see new shifts, but we lose others. So if we walk through this, we see that specific leaf area declines from, this was 70% before, now it's 33%. So this suggests that shifts in trait average are bringing along some amount of shift in trait variability, but still 30% greater variability is not explained. So this means that this is an independent shift in variation after accounting for shifts in the mean. We also see that for trichome density, where we didn't see a difference in variability before, after we account for the mean variance relationship, domestic plants also have greater variability in this physical trait as well compared to wild plants. We find that the variability in chemical concentrations and in chemical richness is a direct product of domestic plants producing more compounds overall, because we see these effect sizes disappear. But we find that declines in Simpson's chemical diversity and also in beta chemical diversity really don't change very much. So this suggests that the process that generates higher overall compound production in plants will not automatically bring along differences in evenness of chemical distributions or in dissimilarity in composition among leaves. So for the second section, we found that variability can shift jointly with the mean and with overall compound production for some traits, but it's often an independent feature of the plant phenotype. So in this last section, we wanted to explore one of the mechanisms that could be shaping the differences in variability we see between wild and domestic plants. So one way that variability could change is if domestic plant leaves are achieving a greater range of traits across ontogeny. So if the actual ontogenetic trajectory of leaves differs between wild plants and domestic cultivars. So our way of approaching this was to analyze the data in a similar way, but separate it by leaf age class. So analyze it within these younger, middle expansion stages and older expansion leaves, more expanded leaves. So we expected that these effect sizes should be much smaller within than among leaf age classes if shifts in variability are primarily driven by shifts in ontogeny with domestication. So when we look at these results, we find that some of these variability effects may indeed be due to changes in ontogenetic trajectory with domestication. For example, SLA, the effect size declines and is much smaller within than among leaf age classes. These trichome effects disappear entirely, as do Simpson's diversity of chemicals across leaves. So this suggests that for these traits, differences in variability, they're real between domestic and wild plants, but they're generated by differences in leaf ontogenetic trajectories. And in contrast, for other traits, such as for leaf water content, and again, for beta chemical diversity, we find these persistent shifts in variability, and they're of similar or even greater effect size than when we quantify them across leaves of all ontogenetic stages.
4 minutes left.
Thank you. So this suggests that domestication can shape differences in variability via multiple mechanisms. So they can change, domestication may change the range of traits achieved across leaf ontogeny, or it may be more pervasive and shift variability independent of leaf ontogeny. And one reason we think this could be really ecologically interesting is that we know herbivores can use leaf age and perhaps nodal position as a quality cue. So this suggests that leaf age or nodal position may be more or less noisy as a foraging cue, and also that shifts in variability may be difficult for herbivores to avoid, even if they specialize on a particular plant tissue age for certain traits. So if we go back to our big question of, has domestication selection altered levels of trait variability and diversity within plant individuals? Although this is a very early step, I hope I've convinced you that yes, this particular axis of the plant phenotype may be underappreciated when we think about domestication selection, and even beyond domestication selection in natural systems as well. And this trait manifests in complex ways across leaf traits, and there are many open questions with regard to both cause and consequence. But we know that trait variability can be as, if not more important than trait means for a range of ecological processes. So this suggests to me that this trait, this within plant variability, may be a novel frontier of plant resistance or susceptibility in crop systems. And when it varies independently from the mean, it may have the potential to be manipulated independently through crop breeding or simply as an independent component of the plant phenotype. So thank you for listening. Here's my contact information if you have any questions I can't get to in the couple minutes that remain. And I'll just give a quick shout out to my advisor, Will Wetzel, who will also be discussing variability later tonight at 6:20 at a very different scale. So check that one out as well.
Great talk. I'll wait for, we could probably fit at least one question in if people want to throw it in the chat or the Slack. Okay, from Vince Formica, do you think this is special effect from domestication, or do you expect to see this in any strong selection scenario?
That's a great question. I think I would expect to, so in the case of alfalfa, I think it's an interesting crop system because what we've probably been selecting for are fast growing plants that can produce big leaves for our cows to eat. So we're not selecting on a harvested organ that we harvest individually. So I think we would actually expect this in any plant where we see kind of a growth defense trade-off or a tolerator plant that puts a lot of effort into plant growth and may produce a lot of secondary compounds or produce really big structures. Then I think we would expect similar shifts in variability, both tied to those shifts in means, but also potentially independently.
Great. To avoid running over, I'm going to let you answer maybe the other two questions. We have at least two more, one from Becky and Jen, and I'll add my own as well.
Sorry for going long. Thank you.
Oh, no, that's what the chat's for.
No, we're right on schedule here. So our final speaker before we'll have a short break is Dale Clement, who's going to be talking about evolutionary history and effects of within generational and transgenerational plasticity on fitness after rapid environmental change. So I'll turn it over to Dale.
Let's see, Mariah, you might still be sharing your screen.
Oh, sorry. Thank you.
Oh, okay, thank you. Okay. Okay, so my name is Dale Clement. I'm a PhD candidate at the Center for Population Biology in the University of California, Davis. And I'm going to be presenting on a modeling project I've been working on that ties the evolutionary history of a population to the effects of within generational and transgenerational plasticity on that population's fitness after rapid environmental change. But first, I'd like to thank my collaborators and co-authors, fellow graduate students Isabel Nalon and Nicholas Roberts, as well as Sebastian Schreiber, Pete Trimmer, and Andy C. So human-induced rapid environmental change is ubiquitous in the natural world, from increases in temperature across the globe to a greater frequency of extreme events to the spread of invasive species. A key question then is, when can natural populations cope with this human-induced rapid environmental change, or HIREC? One way in which they can do so is if they possess pre-existing strategies for dealing with environmental change due to having evolved in variable environments, and can then utilize these pre-existing strategies to cope with human-induced environmental change. Of course, this means that a population's historical environment determines whether or not these pre-existing strategies have evolved within the population. So what I'm going to do in this talk is present a theoretical framework that draws a through line from the historical environment in which a population evolved to the adaptive strategies that are expected to evolve in that environment, to how well those adaptive strategies fare in allowing populations to cope with human-induced environmental change. And then, because this is all very abstract, I'll try to make the results a little more concrete by focusing in on two, if I have time, biological scenarios. The first is a population of prey that must use anti-predator defenses in order to cope with a novel predator. And if I have time, a flowering plant that must adjust its phenology in response to increased environmental variability and the decreased reliability of environmental cues. So when I talk about an adaptive strategy, I'm specifically talking about three things for the purposes of this model. First is within generational plasticity, or WGP, which is the dependence of an individual's phenotype on the environment that it experiences during its own lifetime. So this includes developmental plasticity and phenotypic flexibility. Then there's transgenerational plasticity, or TGP, which is the dependence of an individual's phenotype on the environment experienced by previous generations. So there are a variety of mechanisms by which this may occur, including behavioral mechanisms like parental investment or parental habitat choice, as well as genetic mechanisms or physiological mechanisms. And then finally, there's bed hedging, which is a strategy by which an organism lowers its expected fitness in order to reduce the variation in fitness experienced by it or its offspring over time. So the framework that I'm using builds on that of John McNamara and others in a 2016 Ecology Letters paper that treats these three components as part of an integrated system that I refer to as the phenotypic determination system, by which information about the environment or environmental cues are integrated into an individual's phenotype. So then WGP becomes the pathway by which cues about the current environment are perceived by an individual and incorporated into that individual's phenotype. TGP, transgenerational plasticity, is the pathway by which information about previous environments are perceived by individuals, passed from parents to offspring, and then incorporated into the offspring's phenotype. And bed hedging represents, in some sense, the absence of environmental information, where offspring phenotypes have a randomized component. So one question that naturally arises is, if individuals have access to current environmental information, why rely on any other cues? After all, transgenerational plasticity necessarily conveys information with a one-generational delay, and bed hedging represents the absence of information. But the reason why you'd rely on either TGP or bed hedging is because environmental cues are not perfectly reliable. So in some cases, if current information is inaccurate, it may be better to rely on slightly out-of-date information than unreliable, up-to-date information. And then finally, the phenotypic determination system is defined by Q weights, which is the weight placed on each of these three components. And this represents the levers by which natural selection can adjust the phenotypic determination system and allow populations to adapt the phenotypic determination system to particular environments. So then the key question is, does the phenotypic determination system that was adaptive before environmental change in historical environments still produce successful individuals after human-induced rapid environmental change? And I answer this question in two steps. First, for a given environment, we can calculate what the optimal set of Q weights are for that environment. So what is the best strategy in a historical environment? And if we know what the best strategy is in the historical environment, does that previously adaptive strategy allow populations to persist after a rapid environmental change? So going into the results, this is before environmental change, so relating the historical environmental conditions here, environmental variance on the horizontal axis, to the phenotypic determination system, here Q weights on the vertical axis. We see that whenever within generational cues are reliable, we have within generational plasticity increase as a function of environmental variance, transgenerational plasticity decrease as a function of environmental variance, while bed hedging does not appear. On the other hand, whenever within generational plasticity is unreliable, first we see the same general patterns for WGP and TGP. WGP increases, TGP decreases, but now we see bed hedging for high levels of environmental variance. So then we can summarize these results by partitioning the parameter space of historical environmental conditions, here environmental variance on the horizontal axis, and reliability of within generational cues on the vertical axis, where when environmental variance is low and within generational cues are reliable, you see moderate levels of both WGP and TGP. When within generational cues are unreliable, we see low WGP and high TGP. Conversely, when you have high environmental variance, you see low TGP and high WGP. And then when you have WGP is unreliable and the environmental variance is high, you see moderate within generational plasticity as well as bed hedging. So then there's the question of why we see these particular patterns. Is there something about this informational way about thinking about these mechanisms that allows us to figure out why this is the case? And the answer is yes, because thinking about environmental variance, if TGP represents information that is slightly out of date, the greater the variance in the environment, the greater the difference there is between past environmental conditions and the current environment. Whereas if we think about within generational cues or information about the current environment as some representation of the true environment plus an amount of noise, then as the environmental variance increases, the signal to noise ratio of within generational plasticity also increases. So within generational plasticity becomes more reliable. And this is particularly interesting. Of course, then you also rely on bed hedging when none of the cues are reliable. And this is particularly interesting because this pattern of an apparent negative association between WGP and TGP across these environments has some empirical support. So it'll be particularly interesting if this proves to be a more broad phenomenon that would provide support for this theoretical result. Okay, so now going into the different types of environmental change. First, I'll look at a shift in the mean environment, which is the most commonly studied type of environmental change, but also I'll look at an increase in environmental variance, where the spread of environmental conditions increases after the environmental change. And this is particularly a topic of particular interest in the context of extreme events, where you have a greater frequency of environmental conditions on the tails of this distribution. And then finally, I'll look at an increase in within generational cue error. So before the environmental change, the observed cue more or less matches the true environment. But after the environmental change, we see that the observed cue is no longer a good indication of the true environment. So what makes this approach particularly powerful is we can take these qualitative results from about the historic environmental conditions and then superimpose a graph of fitness loss after an increase in the mean environment, thereby mapping from historical environmental conditions to adaptive strategies to fitness loss after environmental change. Here, fitness loss is high for tan and green colors, meaning that populations do poorly, and for blue and purple colors, fitness loss is low, meaning that populations cope well. And what we see is that fitness loss is highest whenever the environment is the most stable. Not shown in this figure, but we also observe that when the mean environment increases, environments that are uncorrelated across time also have populations that do poorly after this environmental change. And the reason for this is that populations do poorly with respect to an increase in the mean environment when total plasticity is low. So going on to an increase in the environmental variance, we see that fitness loss is highest precisely in the area where transgenerational plasticity is favored. And this makes sense because an increase in the environmental variance makes TGP less accurate. Conversely, fitness consequences for an increase in the within generational error is highest whenever within generational plasticity is most favored in the historical environment. So this leads us to the general observation that populations do poorly whenever previously reliable cues become unreliable due to environmental change. So to summarize the results that I just presented in key points, we see a negative association between WGP and TGP across environments. Fitness loss from a change in the mean environment is greatest when total plasticity, both WGP and TGP, is low. And then fitness loss from other kinds of hierarchy is greatest when reliable cues become unreliable. Now, in the several minutes I have left, I'm going to help contextualize these abstract results in the context of a population that must rely on anti-predator defenses in order to cope with the invasion of a novel predator. So the first step in going from abstract to concrete is figuring out what types of hierarchy are represented by this scenario. So if you have a novel predator, assuming the rest of the community doesn't change, then you would see an increase in the mean predation risk. And because the predator is novel, the cues that the prey species relied upon in order to recognize the predator may not work. So you might have a decrease in the reliability of the within generational cue. So given these types of hierarchies, we know what strategies cope well. So you want high levels of plasticity to cope with an increase in the mean environment. And you want low levels of WGP to cope with a decreased reliability in within generational plasticity. And this corresponds to historical environments where environmental variability is low and the within generational cue is inaccurate, because these environments promote transgenerational plasticity, which is the strategy that copes best with these particular types of environmental change. So I'll skip my second example, but I'm happy to go back to it if you have questions about it. So where do we go from here? There are several very pressing questions that I'd like to tackle in future research. One is, what if the cue integration system is able to evolve after environmental change? So then this becomes an evolutionary rescue scenario with both WGP and TGP, which I think is particularly interesting. Also, there's the question of what happens if there's a cost to plasticity? And this is one thing that I'm currently working on. Well, I am working on with collaborators, and I'm happy to get into this if you have questions about it. And we are also interested in what if you have interactions between TGP and WGP? So it's not just linear, but TGP may be able to prime individuals so they respond with greater within generational plasticity. And finally, I would like to acknowledge once again my co-authors and my funding sources, particularly the NSF for a whole slew of grants, the German Research Foundation, and the University of California, Davis. And now I'm happy to take questions.
Great talk. I'll wait for questions in the chat or Slack. First is from Samuel Shiner. Does your model include an explicit genetic component?
Yes. So I de-emphasize that in this talk. There are actually, so this following the McNamara framework, there are actually two ways in which you can speak of genetics. One is rapid evolution as part of this broader phenotypic determination system, where populations, where the phenotypes are dependent on a genetic cue that can evolve in real time. And then the other sort of genetic component are the cue weights, which represent a more long-term genetic component that's assumed to have limited genetic variability. So we did include the genetic cue or rapid evolution in the model, but for the parameters that we looked at, it ended up not mattering so much. And the reasoning for this is essentially that evolutionary rescue or rapid evolution tends to matter more when the change occurs over longer timescales, as opposed to an increase in, say, generation to generation environmental variability. And in fact, we do see that when autocorrelation is high and the other mechanisms are somewhat unreliable, we do actually get rapid evolution. And the other time we get rapid evolution is whenever plasticity has costs. And we're planning on writing a whole other paper about that.
Great, we probably have time for one more question before break, if anyone has one.
I think there, oh, never mind.
One new one, presumably species with different life histories For example, generation time, end up with different WGP, TGP rates.
Yeah. So this isn't something that we explicitly considered in the model because it's assumed to be simul pares organisms where they live for one sort of time step and then they're replaced entirely by their offspring. But one way in which you can think about this, and there has been some previous work on this, is the timescale of the environment being faster for more long-lived species. So then you'd expect less autocorrelation between environments and possibly higher environmental variability for longer-lived, as experienced by longer-lived species, whereas shorter-lived species may experience more autocorrelation. Of course, that depends on the specifics of the system, and a lot of the challenge of getting these abstract results into concrete predictions is fitting the general results into a specific system.
Any more questions before break?
And I'm happy to take any other questions that people have in the Slack, or feel free to email me. I think I put my emails up on one of the slides.
Okay, well, thank you to all the speakers from the first part of our session on trait evolution. We have a 20-minute break scheduled now. I guess that's coffee or lunch or dinner, depending where you are. And we will reconvene at 5:40 p.m. Eastern time, 20 minutes from now, to start the second half of our session. We're probably just about coming up on time. So welcome back, everyone, for the second session of our on trait evolution. Our first speaker is Natasha Gaunaris, whose talk is titled "Feet First: Do Magellanic Penguin Chicks Show Adaptive Growth?" So I'll turn it over to you.
Is it still being cut off?
No. Oh.
Okay.
It's hard to tell from that slide. I think we're good.
Okay, great. Well, thanks for the introduction. Thanks everyone for coming out on a Saturday evening or whatever time it is where you are. I am a faculty member at Gettysburg College, and I conducted this research in collaboration with Steve Boersma at the Center for Ecosystem Sentinels at the University of Washington. So again, today I'm going to be talking about adaptive growth in Magellanic penguin chicks. And adaptive growth basically just has to do with the idea that growing animals will allocate their energy in ways to maximize survival. So Magellanic penguin chicks are long-lived seabirds. They are colonial breeders. They often will come back even to the same burrow or bush nest to breed each year. They're serially monogamous, and they have obligate biparental care. So when chicks are young, mom and dad will trade off every day or two and always be with the chick. And then when chicks are older, both parents will forage to keep up with the chick's energy demands. They lay 2 eggs, and they don't relay eggs when they, if they lose eggs or chicks.
Natasha?
Yes.
We are seeing only part of your slides.
Okay, I'm not sure how to.
It's just cutting off the edges. Probably because your format, I think, is set for the other aspect ratio.
I can try sharing my screen rather than the PowerPoint. That might help.
Yeah, give it a shot.
You'd think after teaching online for this long, I'd be great at this.
Presenter.
Yeah, if you just do slideshow now, I think you'll be good.
Yeah, 1 second. Okay, are you seeing the full slide now?
There we
I
go.
believe,
Yeah.
yep, I think we're good.
Okay. So again, they have obligate biparental care. They have 2 eggs each. They lay 2 eggs each year, and they don't relay. So why I care about the chick growth stage specifically in Magellanic penguin chicks? Seabirds in general have a very kind of quick switch in locomotory mode when they go from chick to juvenile, right? So on land, they need their feet to move around. And then when they become juveniles or adults, they need to either fly or swim, or both. So this is a really interesting kind of question on adaptive growth, because what influences fitness has this really abrupt switch. The other thing is that in Magellanic penguin chicks and a lot of other seabirds, the chick and juvenile survival rates are very low. So in Magellanic penguin chicks, by the time you reach year 1, so the number of chicks returning to the colony as juveniles after the first winter at sea, there are actually only about 5% of the individuals that were hatched that year left. On average, chick survival or chick mortality is about 70%. And that is variable from year to year. So there is this bottleneck that's occurring. And so the selective pressures that are occurring during the chick and juvenile stage could actually influence adult form. So we had 3 predictions for this study under the adaptive growth hypothesis. And we tested these predictions using measurements of 4 skeletal features, so bill length and depth, flipper length, and foot length, in addition to mass. And this was over the course of a long-term study from 1983 to 2017. So we actually had measurements for over 10,000 chicks. So we had about 60,000 measurement occasions, which means all 5 measurements for an age for a chick. We looked at 3 sets of predictions under the adaptive growth hypothesis. I'm going to go through these predictions specifically in order, but I want to give you the overall idea first. So the first set of predictions had to do with growth order and timing. We're going to look at this using growth curves in relation to changes in behavioral status of the chicks over time. The second had to do with variation in shape in addition to size. So does feature size vary only due to overall size in these chicks or also due to shape? And we're going to use principal component analysis to look at this question. And the last has to do with trade-offs between mass and skeletal growth. We're going to look at age-specific allometry and body condition to answer this question. Before I get into these 3 sets of predictions, this is a good time to stop. I just said, of course, that this had over 10,000 chicks involved in this study and started before I was born. So there are a lot of people to thank. Again, Dimorphism has led this study since 1983, and there have been over 30 years worth of field volunteers in the field for 6 months, 14-hour days. So a lot of people to thank for this. And I also wanted to thank everyone who was involved with logistical support and permits and the conference organizers. So the first set of predictions have to do with the order in which chicks grow their features and the timing relative to other events throughout the chick growth period. So here we're going to be looking at everything in relation to to adult size, and specifically 90% of adult size as the benchmark. So I predicted that feet would reach adult size first because they're needed to move around on land, followed by flipper length, and then by bill length or depth. Specifically, I predicted that feet should reach adult size by the time that chicks are left unguarded. So again, larger chicks, both parents need to forage to keep them fed, and so they're left unguarded at their nest, and they'll often move from their nest to avoid predation or to avoid aggression by roaming single males, but then they have to quickly make it back to their nest if their parents come back to feed them. So they don't often move far, but they will move from their nest when they're left unguarded. That both feet and flippers should be fully grown prior to fledging, and there is previous research showing that there is selection on flipper and foot size in this species for fledglings, but not on bill size. So I didn't predict that bill size would necessarily reach adult size prior to fledging. I also had some predictions in the context of other energy intensive processes that these chicks face. So a big one is thermoregulation. They're able to thermoregulate at around 10 to 15 days of age. So I predicted that there would be rapid growth prior to this stage, so that these energy intensive processes of growth and thermoregulation would be temporally distinct to reduce the constraints of energy allocation and demands on parents. Also being larger, it reduces your surface area volume ratio and things like leg muscles can be used, shivering, to help with heat production. And lastly, that most growth would occur prior to the juvenile plumage growth, because that's another energy intensive process that the chicks need to complete in order to fledge. So if we look at the actual results, and here we can see, again, those important events that are occurring that I had my predictions around. So we do see that the growth of all skeletal features and mass are most rapid when chicks are able to thermoregulate. And if we looked at changes in growth, all of these features peak in their slope or change in growth prior to age 15. Next, we see that chicks actually reach 90% of adult size on average of foot growth at age 29 and flippers at age 39. So again, feet are fully grown by the time chicks are left alone at their nest, and flippers are fully grown around the time that chicks start growing their juvenile plumage. And lastly, that flippers and feet are fully grown by the time chicks fledge, unlike bills and mass. So in general, we do see that the order and timing of growth for these chicks does match the selective pressures that they face, both in terms of when they need different features and when energy demands are highest for things besides growth. The second prediction has to do with variation due to shape across chicks. So we were interested not only in how overall size varied across chicks, because we know that the growth rates do vary quite a bit, but in terms of how the ratios between features vary. So when we're looking at principal component analysis of skeletal features, we'll often assume that first principal component basically represents overall size, because all of your features are increasing with overall size. So they're all in the same direction, all have high loading values. So here, our first principal component is our size component, and then higher order principal components tell us something about how the ratio of features varies across individuals when size is accounted for. We predicted that over time, the variation due to shape would decline. Basically, that chicks would forage or would fledge at a specific shape. So there would be this canalization of a specific shape that's ideal for foraging due to things like drag and wing loading constraints. So now I'm going to look at what we found for the results. So here we're looking at the variance described by each principal component. And again, this is a principal component of skeletal feature, principal component analysis of the 4 skeletal features that we mentioned before. So as you can see, the variance explained by shape, so the variance not explained by the first principal component, is highest in just hatched chicks. So in just hatched chicks, that's actually the only time that the second principal component is above the threshold that's considered significant based on the broken stick model. However, variation due to shape decreases rapidly as variation due to size increases while chicks are rapidly growing. And at fledging, there's a little bit more variation due to shape. It still is not above that broken stick model threshold. And based on what the second principal component signifies, we believe that this is due to sexual size dimorphism. So at fledging, males are slightly bigger than females, but they're particularly large in their bill length and depth. And what we do see is that the second principal component is a trade-off between bill size and flipper and foot size. So this in part matches our predictions, right? We do see a decline. We do see a increase in variation due to size and decline in variation due to shape at first, but then we do see a slight increase in variation due to shape, which might be a result of sexual size dimorphism that increases throughout the chick growth period. The last set of predictions we're going to talk about are the predictions of mass versus skeletal growth, so how those two energy demands are balanced in growing chicks. And here it's important to know that most chicks in this species die of starvation, and that starvation rates are highest during the first 10 days of growth, but in general are high during the first 30 days of growth. So what we predicted here is that higher rates of starvation will lead to prioritization of fat or mass, and lower rates of starvation will lead to prioritization of skeletal features. So later in growth, they're also trying to get ready to fledge, so they have to make sure they're that certain skeletal size and shape. So what we're going to look at for the results, for the first set of results, is a principal component analysis across size that we use PC1 as an indicator of overall body size. And then we regressed mass on PC1 to look at the body condition of chicks throughout time. And what we found is that in general, the residuals, so the body condition index, are highest in these young chicks. So they increase quite rapidly, indicating that chicks are trying to improve their body condition or having a higher mass to skeletal feature ratio as they age in these first 10 days. And then they decline slightly. So this in general matches our prediction that younger chicks, when the starvation rates are highest, are going to try to keep some fat reserves, and older chicks are going to prioritize skeletal feature growth and have a lower body condition index. The one thing I wanted to point out here is that at each age, you do see that there's quite high variation in the residuals. And so we wanted to look a little bit more at day-specific growth and allometry. And specifically, we were interested in the question of whether chicks that are small for their age prioritize their energy differently than chicks that are large for their age. This is a relevant question because the chicks can vary quite a bit in size at a given age. So these are two chicks from the same nest, a first hatched chick and a second hatched chick. So they probably hatched 1 or 2 days apart. So you can see that within 1 or 2 days of age, these chicks can be quite variable in their size. In this case, mom and dad are really focusing on making sure chick number 1, first hatched chick, fledges, and probably not feeding chick number 2 very much. So there are basically 2 ways that you can think of this. One is that individuals will prioritize skeletal growth when their resources are limited. This is the lean priority model, and it's generally used when, or thought to occur when food resources are relatively predictable on a day-to-day basis. The fat priority model, chicks or animals, individuals will prioritize fat reserves over skeletal growth. So when resources are limiting, they will try to store fat because they want to avoid starvation if they don't get fed for a while. So with the lean priority model, we would expect that light chicks for their age would have positive residuals. So they would have a high skeletal size to mass ratio. And of course, there are physiological limits to growth. So we do predict that the largest chicks will actually show negative residuals. So they have to, at some point, they can't grow any faster skeletally, and so they will have a high or a low skeletal to mass ratio. In the fat priority model, the smallest chicks, the lightest chicks, will have a low skeletal growth to mass ratio because they will be prioritizing their fat reserves.
4 minutes left.
So here, we're going to actually predict a lean priority model because chicks, although there are these high starvation rates, seabirds are foraging at relatively predictable fronts, unlike something like an insectivorous bird where things like wind conditions can vary day to day. They do have these fronts that they're foraging at that are seasonally relatively predictable. So day to day, predictability shouldn't vary very much. And the chicks are also being fed very frequently when they're young. So here I've just selected a few ages to show these allometric relationships. And what you can see is particularly in the younger chicks, skeletal growth is being prioritized in the lightest chicks. We don't really see any evidence that there are physiological limits to growth being reached. We can also look at this across ages. I'm not going to spend much time here since we're running out of time, but I had just selected a few ages so you could compare that to the linear allometric growth. So in summary, the growth patterns of these chicks closely track the selective pressures and behavioral states that they went through as they grew. that chicks varied in size but showed little variation in shape, and that variation in shape declined rapidly in young chicks, then increased slightly prior to fledging, possibly due to sexual size dimorphism. And that young chicks prioritized mass on average, but the lightest chicks of each age prioritized skeletal growth. So they followed this lean priority model. With that, I'll see if anyone has any questions. Sorry about the rough start there.
Great talk. It's still coming in right on time, even with the technical difficulties.
I just had to talk New York style, which means double speed.
And we probably have time for one question, if somebody can put one in the Slack or chat. If people don't get to type them in time, this is the benefit of Zoom. I'm sure you can just respond in the chat or the Slack during the next talk.
Great. Thank you.
Thank you. What a cool data set.
I'll wait a little bit just in case there's anyone coming in. We're about a minute early, I think. Kelsey, if you want to get your screen share up to make sure it's good.
Good call. Let me do that. Come on, Zoom. What are we doing? Hopefully that looks all right.
Looks good to me.
Cool beans. Thank you.
Just give it 30 more seconds.
That was great.
Okay, so our next talk in the second half of the trait evolution section is by Kelsey Byers, titled How Does Floral Scent Change as One of the World's Largest Flowers Opens? Looks like we'll get some good pictures. Turn it over to you.
Cool, thanks. So in contrast to the previous talk, which had a truly impressive time of sampling and also sample size, this is a story about a sample size of 1 and a time of sampling of approximately 17 hours. So I'm afraid that this is going to be rather more of an anecdotal story than a statistically testable story, so to speak. My contact info is on the bottom left of the slide. On the right side of the slide is a picture of me 10 years ago when I was a wee grad student, sampling from Amorphophallus titanum, which is the world's largest unbranched inflorescence. It's actually composed of a large number of much smaller flowers, but to the layperson, we would consider it one of the world's largest flowers because it truly is gigantic. So I want to start out by acknowledging the folks that helped me during my PhD and helped me think about this Amorphophallus tiny sub-project. In particular, the Riffle lab, Jeff Riffle in particular, as well as the Bradshaw lab, and in particular for this particular talk, the biology department greenhouse staff who helped rear these plants to maturity and in particular helped foster the blooming of this one Amorphophallus titanum. So I'm a chemical ecologist primarily, and I think about chemical ecology as a window into biotic interactions. So the International Society for Chemical Ecology has defined chemical ecology as an understanding of interactions between organisms and their environment. That's one view of ecology. The question below being, how does the bee interact with the flower to lead to pollination? And the definition continues, that are mediated by naturally occurring chemicals. So in this case, the flower is emitting scent, the bee is sensing the scent, and that attracts the bee to the flower, which leads to pollination. So one of the things as human beings is that we tend to be a little bit visually biased. We look at things that are colorful or things that are large and impressive, and we sometimes forget about this invisible hand of chemistry when it comes to interactions. But that is changing, which really brightens my heart. And this particular example I'm going to talk about is one of the more obvious examples of chemical communication. So many different factors can influence pollinator and other organismal attraction to flowers, such as shape, pattern, color, texture, size, and of course, scent. And for this talk, I'm going to focus particularly on scent. As I mentioned, this is a system that is very well known for its scent. So in 2011, a so-called Torch in the Forest opened in Seattle. There were many news reporters, as you can see on the lower left. Here's a picture of the flower opening and a giant putrid, quote unquote, corpse flower blossoming at the University of Washington greenhouse. And largest, smelliest, etc. This is a flower that basically you can go onto Wikipedia and get a list of all of the times that it has publicly bloomed in various glasshouses around the world. Because it opened up during graduation weekend, we actually had about 3,000 visitors coming to view the plant in bloom in 2011. And for those of us who work on plants, sometimes you feel like you don't get as much public attention, but we got plenty of public attention during this particular event, which was great. So what did the plant actually smell like? So I've heard from people who smelled multiple of these that they do smell a little different each time. My subjective scent description was that it was the best camembert cheese combined with baby diarrhea. It's a very, very stinky plant. So what do we know about the life cycle of the Titan arum? Essentially, it has a corm. Most years, the corm grows into this huge leaf with complex leaflets, as is depicted in this diagram by the Chicago Botanical Garden. And then once the corm is heavy enough, it actually produces a flower. The flower is huge. It smells like a dead animal. It attracts pollinators. It's a brood site deceptive. So basically it says, I am stinky and I am warm and I am colored like a carrion corpse. Come and lay eggs in me, which is to say, come carry my pollen away. It's worth mentioning that despite this massive investment in this particular inflorescence, which can be several meters tall, the flower actually withers within a few days after it opens. So it has to produce leaf for multiple years in order to build up enough storage just to produce this flower that really doesn't last very long. So it's a protogynous flower. The smell aligns with the female phase. So here's a cutaway of the same flower a few days afterwards, after it opened. And you can see the male flowers at the top and the female flowers are at the bottom in red. The spathe, which is sort of the part that surrounds the central column of the flower, of the inflorescence rather, opens in the afternoon and evening and it remains open during the female receptive period. Then the female flowers stop being receptive as the spathe wilts. The male flowers then release pollen. The pollinators who've been duped into visiting fly away. The main pollinators I hypothesized to be carrion beetles and flesh flies, which are attracted again by the combination of scent and temperature and color. But despite the fame of this flower and the fact that it becomes the front page article on global news when it blooms, we actually don't know very much about it in terms of its natural history. One of the reasons for this is that the natural environment is on some islands in Indonesia, and it's not very commonly seen in its natural environment. So most of what we know about this plant is based on individual examples in glasshouses around the world. It's worth mentioning that floral scent release has been reported to be during the first night only, which is the female phase. So in a prior study in 2010, the year before this particular plant flowered, a prior temporal analysis was done and looked at temperature and odor description. So on the right-hand side in part B of the figure that I'm showing here from Shirasu et al., they're actually plotting the spadix temperature, and they're showing in the two pictures in part C the fact that the spadix actually gets very, very warm during the blooming period. On the left-hand side, they're depicting the quality of the odor to the human nose. So a slight rotten fruit-like odor at the very beginning, then it starts to smell like rotting animals, then it starts to smell like rotting fish. And the darker the gray in that part A, the stronger the scent is to the human nose. But this isn't actually a quantitative description. This is just a qualitative description. And that's probably because scent is hard to measure, especially scent on such short time scales. You really need to have specialized equipment. And fortunately, we happened to have access to that equipment. So the question was, could we get a quantitative idea of how the scent actually varies over time? So my advisor was actually away and he calls me on the phone as I'm on my way to work. The plant's about to bloom, the media are coming. Let's do some science. So we use solid phase microextraction or SPEME in order to extract the scent from the plant. So it's one standard way of capturing floral volatiles. Basically, you mount a fiber, and in this particular case, we mounted it next to the spadix. So that's the fiber being enclosed in a holder there, and the arrow is pointing towards the actual fiber. So volatiles in the air actually adsorb onto the fiber, and then we immediately run as soon as the sample is done, place the fiber in a gas chromatography mass spectrometry machine, then that heats up the fiber. The volatiles that are adsorbed onto the fiber then go into the machine and are analyzed. It's worth mentioning that normally when you do this, you would enclose your flower of interest in a bag, some sort of device to sort of concentrate the scent within that area. This flower is too large to put in a bag, and also 3,000 people would be very disappointed if the flower were in a giant bag. But fortunately, it smells so strongly that it was possible to just mount the fiber next to the spadix. So I used SPEME, as I said, to collect volatiles from next to the spadix. I collected for 30 minutes, approximately every 1 to 2 hours, depending on how asleep I was. So I tried to do it every hour. There were a couple of periods where I was deeply asleep and my alarm didn't wake me up. It's worth mentioning I swore at the start of grad school that I would never spend an overnight in the lab, and I broke it twice, and this was one of the 2 events, and it was definitely worth it. So I collected scent 15 times, starting at 7:04 p.m. the evening that it opened until 3:52 p.m. the next day, when scent had essentially disappeared from the flower relative to the background in the greenhouse. I then ran the samples immediately on the GCMS to analyze the sample and clean the fiber for the next extraction. So I was running back and forth between the lab and the glasshouse all night. And of course, one of the cool things about thermal desorption is it allows identification of these low molecular weight compounds that otherwise would be lost in a traditional solvent extraction, because on the gas chromatography system, they would be eluting at the same time as the solvent peak and they'd be washed out.
Shall we take the frittata out of the refrigerator?
Sorry, someone's not muted. Okay, thanks. So what major compounds are present in the scent? I identified a total of 12 main volatiles, identified just by library hits only. So this isn't based on authentic standards, unfortunately. So 3 of those volatiles are sulfurous, methylthioacetate, dimethyl disulfide, and dimethyl trisulfide. This isn't surprising. Sulfurous compounds tend to smell like rotten meat, and so we're very sensitive to them as humans because they indicate spoilage, and that's part of the reason we respond so strongly to this flower. There's also a nitrogenous compound, some monoterpenes, some other unidentifiable compounds, etc. But relatively speaking, it's a relatively simple scent profile for such a large and complex flower. Several of these compounds that are in blue, including all of the sulfurous compounds, had been previously identified in a couple of other studies that had analyzed the scent of the flower. And the compounds that are in bold, including dimethyl disulfide and dimethyl trisulfide, were larger than 10% of the sample at least one given time point. So there were very obvious differences even looking at the data qualitatively as time went on. So the first trace here I'm showing you is a gas chromatograph output. On the x-axis is time in minutes, and heavier compounds come out later and lighter molecular weight compounds come out at the beginning. And on the y-axis is the abundance, so the bigger the peak, the more abundance there is. So this is at 8:26 p.m., which was the second collection I did. And what I want you to particularly pay attention to in the next couple of slides is the 2 things that are outlined in blue, dimethyl disulfide and dimethyl trisulfide. The reason I'm asking you to pay attention to those is because 1 hour later, you can see there's been a dramatic shift in how much of those scents are being produced. So we've gone from basically nothing to a huge amount of both compounds in only an hour. Then, approximately 4 hours after that previous slide that I showed you, those amounts of those compounds have decreased substantially, although they are still present. And by the time we get to noon the next day, those compounds are essentially absent already. So there's a very, very quick peak of those compounds, and they go away also pretty quickly. The total volatiles vary by about 132-fold over the 21 hours that I sampled. So essentially, here I'm showing you a plot of the abundance of the total volatiles over time, and in gray is the local nighttime. There's a massive sudden increase in scent emission as darkness falls, and that's something that was reported qualitatively by other authors, but this is the first time we've had quantitative evidence of it. The peak emission of volatiles is actually only for about 3 hours. So people would come in the next day and say, oh my gosh, it smells so horrible. How can you be in here? And I'd say, well, look, actually the volatile amounts that you're smelling are many, many fold lower than what we actually smelled at the peak when I was the only person in the glasshouse. And then about 1 a.m. in the morning, there's a sudden drop of emission in scent, going not down to background levels, but getting much closer. This pattern is largely driven in this particular flower by dimethyl disulfide. So you can see that the shape of the peak is approximately the same. And during the period highlighted in the blue box, dimethyl disulfide was more than 70% of the total emissions of the flower. So this particular individual was dominated very strongly by dimethyl disulfide. The other 2 sulfurous compounds that we identified showed similar patterns where they peaked roughly with sunset and a little bit after sunset and then declined in the middle of the night. One thing that's interesting about these 2 compounds, dimethyl trisulfide and methylthioacetate, is that they've been shown to be synergistic in attracting carrion beetles in a study by Trumbo and de Capua in 2020. So basically they put these 2 compounds in traps together, and they showed that basically the 2 compounds found together in a trap were better at attracting these carrion beetles than either compound alone, even when they summed the output of those 2 compounds alone. So there is a synergistic effect of these 2 sulfurous compounds, at least in attracting a potential pollinator. The 2 compounds are really strongly correlated in the time course data that I have. The R value is about 0.8. And their correlations with dimethyl disulfide, which again was that major volatile that we found, are even higher with R values of between 0.86 and 0.9. One of the monoterpenes doesn't seem to vary the same. So monoterpenes are very common floral volatiles. They're things that you would find in your shampoo, for example, things like limonene and linalool. And alpha-theogene didn't seem to vary with the same pattern that the other volatiles did. And that might represent background greenhouse emissions that were captured by the speme. So one of the things I unfortunately didn't do after this study was to take a blank sample of after the Amorphophallus was done blooming. And so I can't actually tell you whether that is background emission or not, but my guess is that it probably is. The other monoterpene that I found, saponine, does agree in its emission pattern with the sort of stinky sulfurous volatiles where it peaks overnight and then declines relatively rapidly in the middle of the night. So the scent emission coincides with the published thermal temporal data. So on the top here from Barthelot et al. In 2008, we're seeing the spadix temperature. And on the bottom, I'm showing you the total floral scent emission, the graph that I showed you earlier. And the two blue lines indicate the two spots where the time points are the same. And what you can see is that basically when the spadix temperature is the highest, the floral scent emission is pretty much also the highest, and the shapes of the peaks are pretty similar. I did have dinner with some computer science friends the next day, and we borrowed a thermal imaging camera from one of their labs and wandered down to look at the plant about a day after it had its peak emission event. And you can actually see from the color, brighter colors indicate more temperature, higher temperature. You can see that the stem, and in particular the spadix as well, both are quite a bit warmer than the surrounding ambient temperature, although not quite as warm as the human being standing behind them. So just for folks who are curious, this is sort of a picture of the lifespan of the flower. So starting at the very beginning at about 1, 2 o'clock in the afternoon. And the photographs, excuse me, that are highlighted in blue are the ones that I actually have samples during. And one thing that is interesting is that at 5 o'clock in the morning, the upper right photograph, you actually see that the spathe is open to its greatest extent and the color appears to have developed to its greatest extent. But that's actually not when I saw the greatest extent of scent production. The greatest extent was actually to the left at the photo taken at 2054. So there does seem in this particular Amorphophallus specimen to have a slight discordance between the peak opening of the flower and sort of its peak scent emission. But because this is a sample size of 1 and it's not a very big shift, I think it's probably not super significant. So some brief discussion points at the very end of the talk. Despite its huge size, as we know from the past, it only has female receptivity for a single night, and this receptivity aligns with floral scent release. We found out that the floral scent release peaks at sunset and early night when the spathe is near its max size, but not fully at its max size. The strongest floral scent actually only lasts about 3 hours, but we do see a 132-fold change in volatile abundance over a 21-hour sampling period. The scent is dominated by sulfur compounds, which leads to this rotten meat odor, as previously reported. Not all Amorphophallus flowers actually smell like this. Some of them do smell pleasant, but titanium is well known for smelling really bad, and it's because of those sulfur compounds mainly. So just a little lesson, especially for the folks who are still in grad school and still in early research. I just want to suggest that if you see a cool phenomenon like this, if your advisor calls you on the phone and says, this thing is blooming and the media is excited, let's do science, totally do some science with it. If you see this cool phenomenon, measure something fun about it, but make sure that you do the follow-up work to make it rigorous. So I mentioned earlier, I didn't take a blank sample in the glasshouse afterwards, and I do regret not doing that. And I regret not running standards. So do the follow-up work to make it rigorous, even if you have a sample size of 1. And finally, I'll leave you with my contact information in the upper right. I'm happy to take any questions either by email or Twitter or Slack or the chat. Thanks very much.
Great talk.
Thanks.
Folks can drop questions in the chat or Slack.
I will say I would like to have a sample size of more than 1. So if anyone knows of times when a flower is blooming and the appropriate equipment is in the same institution, that would be awesome. Unfortunately, my current institution has the sampling capabilities, but we don't have a glasshouse that has Amorphophallus in it. It looks like there's a question from Howard.
Yes, presumably temp is controlled in the glass house, but variable in the wild. Might this affect timing of events or perhaps not important as plant produces its own heat?
That's a really good point, actually. So this plant did bloom in July, or in June, excuse me, when the temperature control in the glasshouse was actually quite minimal, mostly because it wasn't necessary to maintain the super warm temperatures. But we did have heating underneath the plant. So it does produce a fair amount of temperature. So I think the values are usually up to 5 to 7 degrees Celsius above the ambient. And so it's possible that heating it would have changed the timing of the emission, definitely. But I think the fact that we do see, at least in glasshouses, that coupling of time between scent emission and between temperature is pretty suggestive. Hopefully that answered the question.
Yes, and Christine Russo says you're answering that there have been no opportunities to sample again there or in other gardens.
So there may have been, and there may be other people who have certainly sampled this before. I know, for example, that there's a plant at the Cambridge Botanical Garden that has bloomed. The main problem is finding a person who is interested in sampling and is interested in doing the temporal sampling and has access to the SPEME equipment and the gas chromatography system such that they can run them in the middle of the night when no one else is around. One of the things to know with scent sampling is that a lot of universities operate in these sort of centralized metabolomics cores, which are really great for access to resources. But if you want to run a sample at 3 o'clock in the morning and you have approximately a day's notice that this flower is going to open, that can be hard to coordinate.
And there's another one from Samuel Schreiner who says, in the wild, how is flowering among individuals coordinated?
I have no idea. And to be honest, I'm not sure anyone else knows either. So because, as I mentioned, the flowers are protogynous, there's this issue that they can't usually self-fertilize. Fertilization has been done in glasshouses a couple of times with saved pollen when flowers have bloomed relatively close to each other, but I'm not sure that anyone understands, and I could be missing some data here, but I'm not sure anyone actually understands how they do coordinate flowering together, especially because they live in a tropical environment where there's not like massive temperature cues that could time that. They're not that common in their native environment, and I think there's a lot we don't know about their natural history because they're not so common.
Thanks, I'll stop there so that we can stay on track, but if there's any—
Thanks very much, everyone.
Yeah, maybe you can address them in the chat or Slack.
Happy.
Thanks, Kelsey. So right on schedule. Our next speaker is Wendy Semsky, talk titled Individual Flowering Schedules and Floral Display Size in Monkey Flower, a Common Garden Study. I believe this is a pre-recorded talk, but Wendy is with us and able to answer questions at the end.
Starting the video now, let me know if something's wrong. Sorry, I'm messing something up.
Hello, everyone.
You can't hear us,
My
can
name
you?
is Wendy Semsky, and I'm a PhD.
I've somehow lost my Zoom screen, so I cannot do anything.
We were able to hear that when you started playing it.
I think I can launch it off mine if you want.
Yeah, maybe you can do that.
I've got it queued up if I can share my screen.
Can you somehow unshare me? Because I can't. There we go.
Okay, share screen.
Sorry about that.
Are we good?
There's no audio. But
there's no audio.
The audio's there, it's just really low.
I can turn it up.
I'll try mine again.
Okay.
I know what I did wrong.
Student at the University of Wisconsin-Milwaukee. Today, I'll be
Think we're
talking
good.
about individual flowering schedules and floral display size in monkey flower. Flowering patterns often vary widely within and among populations and can have important consequences for reproductive success. The extent of flowering overlap can influence pollinator attraction, as well as the number and quality of mates. Two important components of flowering phenology are the date of flowering onset and the length of the flowering duration. These figures show flowering intervals for 5 individuals from 4 imaginary populations. The two populations on top differ only in the date of flowering onset, and the two populations on the bottom differ only in the length of the flowering duration. Simultaneous flowering onset results in complete overlap, maximizing mating opportunity since every plant is available for pollination. However, as variation in the dates of flowering onset increases, mating opportunity is more limited. This effect can be mitigated as the duration of flowering increases, leading to increased overlap of flowering intervals. How flowers are deployed throughout the season can also influence pollinator attraction, as well as the extent of among flowers self-fertilization. Plants may differ in patterns of flower deployment, both within and among populations. For example, these figures represent the flowering schedules of 4 plants from 3 imaginary populations. Each row represents an individual, and each circle represents a day on which the plant flowered. The size of the circle corresponds to the size of the daily floral display, so the larger the circle, the more flowers on that day. In population 1, flowering schedules are nearly normally distributed, and plants deploy their largest displays simultaneously. In population 2, however, flowering occurs in pulses, so that peaks do not occur simultaneously. Finally, in population 3, there is very little day-to-day variation in floral display, so flowering is steady over the season. An important thing to note here is that the total flower production does not vary. All 12 of these plants have the same total number of flowers, but they differ markedly in how they are deployed. Although large daily floral displays may enhance the attraction of pollinators and promote pollen export and receipt, they may also experience higher levels of among flowers self-fertilization. This is because pollinators tend to visit multiple flowers sequentially on a single plant. This type of selfing is costly because pollen and ovules that would otherwise be available for outcrossing are instead used for selfing. In populations with substantial inbreeding depression, selection should therefore favor plants that produce small floral displays over many days, rather than plants that produce large displays over few days. One challenge in studying variation in floral display size is that the number of open flowers per day depends on both daily flower deployment as well as floral longevity. In other words, the daily floral display consists of flowers that opened today as well as flowers that opened previously. It can be difficult to disentangle the relative influence of these factors. Approximately 27% of flowering plant species have flowers that last for just a single day. These species can be very useful in understanding how patterns of flower deployment shape individual flowering schedules and how attributes of their flowering phrenology influence total flower production. Monkey flower, Mimulus ringens, is especially well suited for answering these questions. It is a hermaphroditic perennial native to wetlands in North America. It produces purple flowers, each of which lasts for only a single morning,
so floral displays do not overlap between days. Monkey flower is primarily bumblebee pollinated. Every flower produces a fruit, and it exhibits a mixed mating system, so populations differ in their mean selfing rate. In 2018, we surveyed 9 natural populations of monkey flower in Northeast Ohio. We found wide variation among populations in total flower number, which ranged from a mean of 19 flowers per plant to a mean of 85 flowers per plant. There was also striking variation within populations in flower production. For example, in some populations, most plants produced few flowers in total, while in other populations, total flower number varied widely among individuals. And so my talk today will investigate whether this variation in flower production is also present in a common garden, how it is related to other components of flowering phenology, and also how synchronously individuals deploy their flowers across the season. By raising plants in a common garden, we can control for environmental variation and determine whether these differences have an underlying genetic basis. To answer these questions, we raised 20 progeny from seeds collected from the plants measured in our 9 natural populations, taking special care to minimize environmental variation. Seed germination was highly synchronous across populations. When plants began flowering, for every individual, we noted the date of flowering onset as the date on which the first flower opened. Then, every other day throughout the flowering period, we quantified every plant's daily floral display size. And so this means that we tracked the flowering schedule for every individual. We use those data to quantify components of flowering phenology, such as total flower number, flowering duration, and flowering synchrony. In this figure, the x-axis is the date of flowering onset based on the Julian calendar, and the y-axis is total flower number. Each point represents an individual and is colored according to the population to which it belongs. We found that the date of flowering onset is negatively correlated with total flower number. Plants that begin flowering early tend to produce more flowers than plants that flower later, so earlier flowering leads to greater fecundity since every flower produces a fruit. Additionally, we discovered that this pattern is maintained within populations and is significant in most of them, as you can see in the table on the right. This figure shows variation in the date of flowering onset for our 9 populations raised in the common garden. Date of flowering onset varies widely, both within and among populations. For example, in population LNB, the shape is short, indicating that flowering onset occurred over a very brief window. In contrast, in population MCW, the date was much more variable amongst individuals. Similarly, we see in the figure on the right that total flower number also varies widely in the common garden. In some populations, most plants produce relatively few flowers in total, while in others, total flower production is more variable and is evenly distributed among individuals. We also find that flowering duration is positively correlated with total flower number. In this figure, the x-axis is the length of the flowering duration in days, and the y-axis is total flower number. Each point represents an individual and is colored according to the population to which it belongs. Here, plants that flower longer tend to produce more flowers than plants with a shorter flowering duration. This pattern is supported within the populations as well, which I show you in the table on the right. And we also found that the length of the flowering duration is highly correlated with date of flowering onset. And so taken together, these results indicate that plants that initiate flowering early are also able to continue flowering, and this results in greater fecundity. And so now let's take a closer look at the daily experience of plants and how their patterns of flower deployment influence flowering synchrony. Earlier, I showed these figures, which represent the flowering schedules of 4 plants from 3 imaginary populations. As a reminder, each row is a plant, and each circle is a flowering day, so that the size of each circle corresponds to the size of the daily floral display. Previously, I explained how variation, or lack thereof, in the shape of the individual flowering curves contributes to population-level patterns. Now, I want to highlight how daily variation in flower deployment can affect mating patterns among individuals. This idea is best illustrated in population 2, where plants deploy their flowers in pulses so that flowering is more sporadic across the season. As a result, the number and identity of plants in flower differs from day to day, and thus, how synchronously individuals deploy their flowers influences their mating opportunity. We can quantify the extent of flowering synchrony among individuals in each population to determine the extent to which flower deployment varies across the season. This figure shows the flowering schedules for every individual belonging to population MSB. The x-axis is Julian date, so that day 190 is July 9th. Each row represents an individual, so that each circle represents a day on which that plant flowered, and plants are organized along the y-axis by their date of flowering onset. As before, the size of the circle corresponds to the number of flowers in a plant's daily floral display, so the larger the circle, the larger the floral display. In this population, individuals appear to flower sporadically with large day-to-day variation in their display sizes, and the largest floral displays do not occur on the same day. Also, I'd like to point out one day in particular, day 206. What's striking here is that although this day occurs in the middle of the flowering period, only about half of the plants in this population are flowering. This is especially interesting to see in a common garden, since asynchrony is likely to be greater in the wild due to environmental variation. We used Augsburger's synchrony index, which ranges from 0, no synchrony, to 1, complete synchrony, to quantify the extent of flowering synchrony among individuals in each of our 9 populations. And here, I show you the results for just 2 of them. Population MSB, the figure on the left, had the lowest synchrony, while population RIS, on the right, had the highest synchrony. This is especially apparent when we compare day 206 between these 2 populations. As we saw, MSB has only about half of the plants in flower, while in RIS, every plant is flowering. In RIS, flowering peaks appear to be larger, and many plants are quite synchronous in the timing of their largest displays. In this common garden study, we found wide variation within and among populations in the components of flowering phenology, as well as in patterns of flower deployment. Plants that flower earlier tend to flower for longer, which results in greater fecundity. By measuring flowering schedules across the season, we can gain insight into population attributes such as flowering synchrony and the timing of the flowering peak. Thank you very much for listening.
Excellent talk. All right, I'll wait for questions to emerge in the chat and Slack.
I think there is one in the chat.
Okay, does the greater fecundity of plants that flower early carry through to increase seed acceptance from Kelsey?
Yeah, you can hear me and see me, right? Just want to make sure. Okay, awesome. Yeah, so that's a really great question. However, I have not yet gotten through all of my seed counting.
So I am going to find out that question, but I don't yet know. Yeah, however, we do know that in the wild, seed set is highly correlated with total flower number. And so I suspect that since the plants that have that do flower early do produce more flowers in total, I suspect that that's also going to be true for seed set as well.
Great. And Jen Coughlin says, super neat. Do you know if any attribute of the flowering display was correlated with mating system variation across these populations?
Yeah, good question. So I did do another study that same summer that used siblings of the plants that I've used in the study that I talked about today. In a second study where we established experimental arrays in a common garden in the field where pollinators were visiting naturally. And so I'm going to genotype the progeny from that experiment and compare the size of the daily floral displays with the population selfing / outcrossing rates. So that's another question I'm interested in finding out, but I don't have those data yet.
And final question from Ava Fisher. Great talk. Given the flowering earlier appears associated with increased fecundity, what forces do you think prevent all populations from forming that strategy? Why is selection not clearly directional?
Yeah, that's a good question. So sorry, I'm just rereading the question to make sure I understand in the first place. Yeah, so one of the things that could be affecting selection on flowering time would be differences in pollinator visitation rates. So there could be differences in whether flowering early is associated with the timing of the pollinator visitation. And then also there could be herbivore pressures that are affecting selection. And so plants that are flowering earlier may either be able to avoid herbivore pressure later in the season, or conversely, if herbivores are active early in the season, then by deploying most of their flowers later, they can avoid that in some way. So that's not an exhaustive list, but those are two ways that could happen. And so, yeah, those are really interesting questions.
I think we're gonna have to leave it there. Thank you. The questions can continue in the Slack. Our next talk is by Stephanie Aguillon on leveraging extensive hybridization to identify genes related to coloration differences. I'll turn it over to you.
Great, thank you. Does everything look okay on your screen?
Looks good to me.
Wonderful, great, thank you so much. Great, so I'm really excited to be talking to you today about some of the work I've been doing for my dissertation. This is my first time at a standalone ASN meeting, so I'm really excited and very thankful that you all decided to do a virtual meeting, and I'm really appreciative of all of the work that all of the volunteers have been doing to get this meeting up and running. So I am really interested in coloration and thinking about the genetic basis of coloration, and I think about this a lot in birds in particular. As you can see here, birds are, I think, a really great study system for this. And in particular, what you might notice when you look at a lot of different birds is many different coloration patterns across their body, many different patches that differ. Some birds have whole body coloration, others have very small regions of the body that differ in coloration. And one of the things that has been really taken advantage of in birds, and particularly in the last 5 to 10 years, is systems that have low levels of background divergence. And so this has been done in a number of systems, and I'm just showing you here a few of the studies that have come out. And these systems have low levels of background divergence for a number of different reasons. Sometimes it's hybridization or a hybrid zone, sometimes it's artificial selection. So a lot of this work has been done particularly in canaries in the lab, could be recent or rapid radiation. So it doesn't really matter what results in the low levels of background divergence, but one of the things that's really useful in these studies is having that low level of background divergence. And for me, in thinking about the genetic basis of coloration, I'm particularly interested in thinking about hybrid zones or hybridization in general. And what you might have noticed, or maybe not, depending on how much you know about birds, all of these individuals that I showed on the previous slide are hybrids or systems that do hybridize. So using these individuals is of great interest to me and is the thing that I've been focusing my PhD on. And in particular, I've been focused on the northern flicker. This is a really common woodpecker that's distributed across North America. So the range is quite large, and it's divided in this western-eastern way. So in the west, there's a red-shafted flicker. In the east, there's a yellow-shafted flicker. And the reason that we know that these birds hybridize, and they hybridize in this broad region here in orange, is because they differ in a clear, they have clear phenotypic differences. And hybridization has been known in the system for a really long time. It was first recorded, to my knowledge, by Audubon in the late 1800s. And the reason he knew that these birds hybridize is because they look pretty distinctly different. So when you look at these birds, and I'm showing you here 2 illustrations, very clearly they differ in the color of the wing and tail. So they have this bright salmony red color versus this really bright yellow. That's very obvious. And that's what they're named for, of course. But if you zoom in on the head and face, which you might notice if you look closely, is they also have a number of differences there as well. So if you have this bird in the hand, or if you're looking at a museum specimen or through binoculars, it's really possible to tell that you have a hybrid or not. So I'm just going to run through the differences that they have and that I'm going to talk about later, but know that you don't have to keep them too much in mind. So they have this difference in the male malar stripe, which is this mustache stripe, red versus black. They have differences in the throat color, which you might notice is that they sort of differ in like the colors at the bottom of the face versus the crown, and that sort of switches between the 2 different subspecies. Also the ear coverts, the crown color, as I mentioned, and then they have this broad red patch on the nape of the neck in yellow-shafted individuals that's absent in red-shafted individuals. And so they have all of these phenotypic traits that differ between them. So it's easy to tell if you have a hybrid. And that's because these traits mix and match. So you can have a variety of combinations of these 6 traits in an individual, or you can have an intermediate coloration. So you might have an intermediate wing and tail coloration where you have orange wings and tails. You might have this mustache stripe here on the face that's intermediate. So you have red and black in there. So it's easy to tell if you have a hybrid. And one of the great things about this system, as I mentioned, being very useful, is there's low levels of genetic differentiation, or we expect there to be based on previous work. So this is a system that has had a lot of interest in the ornithological community and the speciation world for a long time. And so in the late 80s and early 90s, there's a lot of genetic work that was done with the idea that these birds look very different. And so we should be able to find the regions in the genome that differentiate them. And using allozymes and mitochondrial DNA at various different scales of sampling, there is no ability to differentiate the taxa at all. So what I did when I started my PhD was use a reduced representation genomic sequencing approach to try to see if we expanded the sampling of the genome, we might be able to tell them apart. And so I was able to differentiate the 2 taxa. So showing you here 2 clustering analyses, PCA, principal components analysis, and structure. I don't need you to take much away from this other than what you should see is the red-shafted individuals grouped with the red-shafted individuals, those grouped separately from the yellow-shafted individuals. So this data set was able to separately cluster individuals, but I still found very low levels of genetic differentiation despite this ability. So if we compare the FST value for individual SNPs, so this is a measure of how differentiated the SNP is, going from 0 completely identical to 1 completely different. This is an allopatric comparison, so birds that are very far away from each other geographically. Looking across this panel of about 9,000 markers, we can see as most of these markers have very low levels of FST, so very, very similar. And in the region of markers that are very different, there's only 7 markers out of about 9,000 that had FST greater than 0.75, no markers that were fixed. So even extending this data set to the whole genome, these birds were still very, very similar. So genetic differentiation, very low. And for all of these reasons, flickers make a really great system for looking into the genetic basis of coloration. So they have these multiple distinct phenotypic differences. They have extensive hybridization in the hybrid zone. And as I just demonstrated, this really low level of background divergence, which you would expect given the increase in sampling extends across the whole genome. So what I'm going to be presenting here today is some work using whole genome sequencing. I'm not going to talk about the methods too much, but I'm happy to answer questions about them at the end of the talk or on Slack later on. But I sequenced and assembled a reference genome and then performed a low coverage resequencing of additional individuals, both allopatric red-shafted and allopatric yellow-shafted individuals. This is to get at a baseline assessment of differentiation, as well as hybrid individuals. And using this data set and the sampling resulted in about 7.25 million SNPs. And that's going to be the data set I'm going to be talking to you today about. So I'm just going to jump right into the results here. So looking across the whole genome, we do find low levels of differentiation as expected, but we do find a number of regions of the genome that are differentiated. So these have high FST peaks. So this is a Manhattan plot here for the allopatric comparison of allopatric yellow-shafted and allopatric red-shafted individuals. The y-axis here is position along the genome. The x-axis is position along the genome. The y-axis is differentiation. So 0 is completely identical. 1 is completely different. And what you notice here is most of the genome is quite similar. So mean genome-wide FST is 0.008, so pretty low, but you do see these peaks. And these are regions of high FST. We have actually able to identify SNPs that are fixed differences between these birds. This is the first time it's ever been possible to do that. We have about 780, which when you're looking at a panel of 7.25 million is very low, but we do have them. And as you might expect, these birds, look very different, but they're very similar on the genomic level. So you might expect that the differences that they do have in the genome are related to the differences that they have in phenotype. And that is the case. So these differentiated regions are associated with the differences in coloration. So I'm showing you here just a PCA plot. All I need you to take away from this is that yellow-shafted individuals and red-shafted individuals sort of separate out on the far edges of PC1. All the hybrids are in the middle. And what you might expect is that maybe PC1 is related to the way these birds look, and that is true. So if you score these individuals based on a phenotypic hybrid index, basically just summing up a score for all of the 6 traits that I mentioned and correlate this with PC1, you see a very strong association. So it seems like these regions of the genome that do differ are the regions of the genome that are related to coloration. And as I mentioned, we have a lot of hybrids in this system. Sorry if my audio just went out. And the hybrids are really useful for genome-wide association studies. And this is what I'm going to be telling you about next. So hybrids have this mix and match genotype with a mix and match phenotype. And we can really use this to our advantage because it gives us some power in these genome-wide associations. So I'm going to jump into the results for those. I'm going to walk you through one result, and then I'm going to sort of contextualize what the findings mean, and then tell you about some more of the results. So we do find regions of the genome associated with coloration differences. I'm going to show you here results for the wing and tail color first. So this is again a Manhattan plot. This time the y-axis though is significance in this association. So the higher up the points are, the more significant the association is between the genomic region and the phenotype. What you can see here is a number of peaks. These are regions that are significantly associated with the coloration of the wing and tail. We can see here we have a number of them. I'm going to show you the results for the other traits as well. But first I'll tell you a little bit about how the results fit into the literature of bird coloration as we know it right now. So we find a number of patterns that have been found in other systems, as well as some patterns that seem to be emerging and patterns that are pretty novel. So there's this pattern that's been found in a number of systems so far that seems to suggest that regions of the genome exert control over very specific patches of a bird's body. So rather than influencing whole body coloration, influencing individual patches, so the colors of the wing or the color of a wing bar. This is a pattern that we find in flickers and has been found elsewhere as well. A pattern that's been found in a few systems so far and that we also find in flickers is regions of the genome that seem to exert control over multiple patches in the body. So these are regions that pop out as significant in multiple associations and seem to maybe control multiple regions in some fashion. And a pattern that we find that seems to be pretty novel, and to our knowledge hasn't really been found or described in other systems up until this point, is a relationship between melanin genes and carotenoid traits. And I'll talk a little bit about that one at the end. But first, I'm going to tell you a little bit about these two patterns first. So looking across the 6 traits, so these are the genome-wide association studies for each of the 6 traits that I mentioned at the beginning. All I want you to take away from this is that 4 of these 6 had significant associations, and I'm going to zoom into these, and that's what I'll be discussing. So across these 4 traits, so this is wing and tail, nuchal patch, this is the patch on the nape of the neck, the color of the ear coverts, the color of the malar stripe, we find some sort of differing patterns that get at these sort of broader conclusions that I just described. So looking across these regions, I've highlighted here in orange regions that are significant in one analysis, but not any others. So these are regions that seem to exert control very specifically over an individual phenotypic trait. As I mentioned, this is a pattern that has been seen in other birds and seems to be maybe a more broader relationship. And then we also see some regions that you might notice here, and I've highlighted it in red, that seem to exert control over multiple patches of the body. These are regions that pop up as significant in more than one analysis, in some cases in many more than one analysis. And so we have some differing patterns here. And then finally, I will touch on this last relationship here that we find, but to do that, I'm going to need to zoom in into the genes first. So I won't belabor this too much because I know tables are not super interesting in presentations, but near the significant SNPs, we do find a number of pigmentation genes. And these are genes that are either known to be involved in pigmentation or have been found in other systems that are suspected to be involved in pigmentation. One of these relationships that's really clear is this gene CYP2J19. This is a gene that was identified a few years ago as being related to yellow to red coloration differences in canaries. This is actually one of the only genes we know at this point that is related to red coloration in birds. And we find that related to the yellow to red coloration differences in the wing and tail of flickers, which is very interesting and is a great relationship to find here. But if you look closer at this table, and I'll highlight this to demonstrate, a number of these traits are carotenoid traits, and a number of the genes are melanin genes. And this relationship seems to pop up multiple times here and gets at this last pattern that I want to touch on briefly. So melanins are these blacks and browns and grays that we see in nature, whereas carotenoids are these bright reds and bright oranges and bright yellows. And to our knowledge, these processes are completely different. So melanins and carotenoids don't use the same pathways, they aren't deposited in the same way. The genes, as far as we know, are very, very different. And so this relationship that we find as being pretty consistent, where traits that are carotenoid based are related to melanin genes, is pretty interesting and is not something that's really been described before. This is something that I'm pursuing further, but I don't have anything else at this point to really solidly tell you other than it's a very interesting relationship and we're not sure why it exists. I will say that this paper is coming out soon in Proceedings B. It was accepted right before the end of the year, and I got proofs, I think on the 29th of December. So I sort of talk about these patterns a little bit more in the paper, but hopefully I'll have a talk at some point following up on what these relationships actually mean and why we find them. And with that, I would like to thank a lot of people that have been involved in this work. So members of the Lovett Lab, as well as a lot of help with field work and museums as well, who provided samples. And then all of the funding sources that I was able to cobble together to be able to do this work for my dissertation. And I would be happy to take any questions. I do have some time, I think, right now, but also later in Slack or email or Twitter or any other approaches. Thanks.
Excellent. I'm gonna have to use my binoculars when I see our flickers in the backyard tomorrow. I think we're solidly out of the hybrid zone, but pretty close in Utah, it looks like. And I'll check for questions in the chat or Slack.
I will stop sharing so I can see you all.
Jen Coughlin asks, first, awesome talk, and do you see if there's an excess of coloration genes on the Z?
Oh, that's a great question. Yeah, so we actually surprisingly don't find a lot of significant associations on the Z, which is very counter to what is found in other studies. The one trait that is different is the Mallar stripe, where we do find a lot of associations on the Z. That is a sex-specific trait, so only males have that expression of that trait, which is maybe one of the reasons. The other potential confounding factor is that we had fewer males, and so that analysis had less individuals in it, which means that the data set is a little noisier. So the follow-up work I'm doing is mostly focused on the other traits where the patterns I think are much, the associations at least seem much stronger, at least for a first pass.
And another one from Liz Tapney. Are there any cryptic hybrids in the system where individuals may not look like hybrids, but they are, say, if they're F2 or F3s, and could that have impacted the results?
Yeah, that is a great question. So hybridization is incredibly rampant in the system. So I suspect most of these individuals are very, very late generation hybrids. So I don't really think any of these are like F1s or F2s. I would say they're very, very backcrossed individuals. Hybridization has been known for a very long time, and it seems like it's been very consistent and very stable in the same region of the continent. But yeah, I think the idea that, you know, we might have more going on here is really important and something that I'm working on pursuing.
Great, we have time for probably one more if someone wants to squeeze a question in at the end. If not, we'll play it safe and we'll let other questions be answered in the chat of the Slack if they come up.
Yep, and I'm
Thanks
definitely
again.
happy to. Thanks.
Okay, so our next talk is by Jill Sanderson on macroevolutionary drivers of complexity in anolis dewlap color patterns. And I'll turn it over to Jill.
All right, thank you for the introduction. So yeah, I'll be talking to you guys today about some of my PhD work, thesis work that I'm currently conducting with Luc Mahler at the University of Toronto. And I apologize if I move a little bit quickly today. We have a ton of ground to cover, so I'm just gonna go ahead and get started. So it's pretty impossible to start any talk about signal evolution without taking a moment to acknowledge the striking diversity we see across species in nature. And one of the critical aspects of this diversity is color pattern complexity, which is something we have a pretty rudimentary understanding of. So why might these signals be more or less complex? Well, there's a few hypotheses about this, and today I'm going to focus on two of them that have to do with sexual selection and signal detectability. So Darwin, I guess, first noticed that oftentimes in one sex, usually the male, they'll have more elaborate coloration and more complex coloration than the other sex. And then Fisher went on to explain how these traits become more exaggerated in systems with higher sexual selection. Other research by people like Johnstone and Muller and Polkinski, they have postulated that these complex colors might arise because of the need to convey multiple types of information about individual quality to mates and to rivals. So there's been a ton of great work on this at the interspecific level, but there's been less progress made at the macroevolutionary scale. In addition to playing an important role in social communication, color patterns also act as adaptations to diverse environments. So much of the research of people like John Endler and Tim Guilford and Marianne Dawkins has focused on how these traits to do with ecological factors will influence the evolution of visual signals and increase their detectability. So I've looking at a couple of these, or most of these, I guess, throughout my PhD, but the ones I'm going to talk about today are ones I have enough data to say something about, and those are size and color contrast. So absolute scale can have a huge influence on how a signal is detected. In Endler's sensory drive paper, he talked about how body size and signal size can really increase the conspicuousness of the signal. And different traits or different aspects of the same trait, for example, size and pattern complexity, can have a huge influence on how the signal is detected, and it can interact with these other factors. For example, we know that visual acuity will degrade at longer viewing distances as well as in dark environments. So in those contexts, it would likely be important for the signal to be larger or to have larger color patches. You can see kind of illustrated on the screen here, we have different visual signals that vary in pattern complexity. So the ones that are easier to detect are going to be the ones that have higher color contrast and are going to be the ones that are larger. So both of these theories provide some insight into the evolution of complex color patterns, but what we don't really know or have less understanding of is the relative contributions of these and how they affect things at the macroevolutionary scale. So to answer this question, I turned to a remarkable study system, and that is the adaptive radiation of anolis lizards. So these guys conduct this really elaborate communicative display that involves the extension of this colorful throat fan called the dewlap. And between species, there's crazy diversity in color pattern, size, and pattern complexity between all different species. So anoles evolve repeatedly and independently in island and mainland contexts. And as such, they also vary in a lot of these ecological features that might predict some of this pattern diversity. So while people often admire this crazy variation in dewlaps and anoles, we have a pretty poor understanding of what actually drives it, again, at the macroevolutionary scale. So one study that took a first pass at this about 15 years ago now used a discrete classification approach, and they found equivocal support for the idea that these dewlaps might be involved in species recognition, but they note that much of this diversity just remains completely unexplained. So although it's a pioneering study, they used a rather simple categorical approach to classify these dewlaps into these various categories. And by doing that, you reduce the amount of power that you have to actually get to concrete conclusions about this diversity. And further, they actually didn't test hypotheses about sexual selection or about detectability. So that leads me to what I'm going to be doing and talking about today. So my first hypothesis relates to sexual selection. And we know in anoles that there's evidence for variation in the intensity of sexual selection between species. So it's possible that these dewlap pattern complexity, it might evolve for signal elaboration via sexual selection. And if so, we would predict that pattern complexity should have a positive relationship with male bias sexual size dimorphism, or what I'm just going to say is SSD. And because we know in this system that SSD is a pretty good proxy for the strength of sexual selection. Another way for elaborating a signal is to make it larger. So one prediction that could arise from this is that species that have large relative dewlap sizes would also have high dewlap complexity. And finally, starting with some of the seminal work of Henry Fitch and Robin Andrews in the 70s, there's a robust body of evidence and research that has went into the idea that there's more sexual selection on island species than there is on mainland species. So as a kind of first pass looking at this at the macro scale survey, we're interested in looking at whether island and mainland species differ in pattern complexity. And the prediction coming from sexual selection would be that island species have more complex dewlaps than mainland species. All right, and my second and no means mutually exclusive hypothesis is that dewlap complexity might evolve for detectability. So in anoles, there's been a ton of work done by people like Leo Fleischman and Manuel Leal and Julian Ning and many, many, many others that has looked at how aspects of the signaling environment relate to how the dewlap coloration is seen in that environment. So a lot of that research is indicated, and as you can see in this figure here from Jonathan Losos's book, that different colors are more detectable in different light environments and against different background spectra. But what's lacking here is there's not much information on how other traits like dewlap size and pattern interact to increase detectability. So for this study, I'm going to be using the comparative method to test how dewlap design patterns scale with absolute dewlap size. And we know that both small and large anoles will use this dewlap as a broadcast signal over relatively large distances. So we think it's meaningful to ask whether absolute size scales with pattern type. And in this case, we would expect that different sized dewlaps would have different kinds of patterns to enhance detectability. So on small lizards like Anolis lyra here, we would expect that they would evolve more bold, high contrast patterns to increase detection. Whereas on larger dewlaps, like that of Megalopithecus, that has more leeway to actually evolve these different designs without sacrificing detectability. And bringing back island-mainland differences again, another major difference between them is that on mainland communities, species exist at much lower densities, and they have to signal at much farther distances. So to aid in signal transmission, we'd also predict that the two dominant colors on the dewlap will be more contrasting on mainland species than in island species. All right, moving right along to the methods. I'm going to go pretty quickly here, but if you have any questions about this after, please do not hesitate to ask me. So I'm going to be looking at this in a sample of 315 Anolis species. And the data I'm using is coming from field collected images. So I know, of course, that images are only an approximation of what the lizards can see in terms of color pattern. But for the purpose of this talk, I'm pretty confident that there's still value in using images as a representation of these patterns. So quickly after starting this project, I realized that the existing spectral and spatial techniques to quantify pattern information really didn't work for anole dewlaps, just because of the sheer amount of ways to get complexity in this two-dimensional design. Therefore, in collaboration with some awesome colleagues, Tristan and Charlotte in the computer science department, we developed these novel tools to measure different aspects of structural information and complexity from images. So those things vary. We look at things like entropy and different kinds of heterogeneity and edginess and all that kind of stuff. But one example that I just popped on the screen here, you can see that Anolis equatorialis has a much higher entropy content in its dewlap than Anolis alli. So we would say that this dewlap is more complex, at least in terms of the entropy axis. Similarly, we also use these tools to segment the image into its two most dominant colors using k-means clustering. And then as a measure of color contrast, we took the average Euclidean distance between those colors. I got size and SSD data from my labmate Ken Twama, measured dewlap size with image J. And to control for the shared history between species in my data set, I'm using Steve Poe's phylogeny. And finally, for those analyses, my main analyses, I'm running phylogenetic least squares regressions. And then I'm sure you can imagine the predictive variables I mentioned, SSD, dewlap size, they're likely highly correlated with each other as well as with body size. So to kind of tease apart some of those relationships, I'm also running a phylogenetic path analysis. And actually, it's using the package of our Zoom sessions tech today. So shout out to Walter for that. So once I have all of these complexity measures on these dewlaps, I put them in a principal component analysis. And very interestingly, I found evidence for two orthogonal axes of complexity. So you can see here, PC1 represents dewlaps with these many little details, or what I'm going to call fine scale complexity. And PC2 represents dewlaps with few bold markings, and that's what I'm going to call coarse scale complexity. So moving right on to the results, first I'll explore the sexual selection hypothesis. So for all the results today, you're going to see two graphs on the screen. So on the left, you'll see the PC1 or fine scale complexity graph, and then PC2, you'll see that coarse scale complexity graph on the right. So as you can see here, the relationship, we find weak but significant relationships for sexual size dimorphism, and complexity. For PC1, it's in that predicted relationship of more complexity, but for PC2, it's in the complete opposite relationship. So this is not what we predicted, and I'll get into a little bit why we think this is happening in a couple more slides. As you can see here, there is no support for the predicted relationship between relative dewlap size and complexity for either fine or coarse scale complexity. And finally, you can see here that mainland species actually show significantly higher fine scale complexity than island species. And we see no difference in coarse scale complexity between island and mainland species. So this again is the opposite of our predictions. So just kind of quickly going back to my sexual selection hypothesis slide, we can pretty confidently say that we reject this hypothesis for evolution of signal complexity in nulls. All right, so moving along to the detectability hypothesis. So we actually find weak but significant results which are in our predicted directions for the relationship between complexity and absolute dewlap size. So I'll just pop some of these dewlaps on the screen here so you can see what the patterns kind of look like. So larger species with larger dewlaps exhibit more fine scale complexity, and then species with smaller species with smaller dewlaps tend to have more coarse scale complexity. So I'll give you a couple seconds to take that in and kind of match the dewlaps up from each of the graphs. All right, I'm moving right along. So you can see here that mainland species actually show significantly higher color contrast than island species. So again, this is just like what we predicted, and it suggests that these traits might help in aid signal detectability. And like I mentioned earlier, I recognize that these predictor variables are likely highly correlated with each other, as well as body size. So here are the results from the path analysis. We have the results for PC1 in this graph here and PC2 right here. And you see the arrows are going to be in the direction of the causal relationship, and the numbers here are the path coefficients or the strength of that relationship. So what we find is that, unsurprisingly, body size has a huge effect on sexual size dimorphism, as well as absolute dewlap size. And any effect that SSD and dewlap size actually have on complexity is likely just an indirect effect of body size. So this would explain why we had the same relationship of increasing fine scale complexity and decreasing coarse scale complexity with SSD as well as dewlap size. And then finally, another major takeaway we get from this is that while body size has partially explains the variation that we see in complexity, we can see the relationship here and here. We also have these unexplored differences between island and mainland species that have a huge effect, especially for PC1, that fine scale complexity, and as well to a lesser degree for coarse scale complexity. So there's lots more to measure here, but we think this may have to do with the population structure, which is kind of corroborated by our findings about color contrast. So to conclude what I've talked about today, this is, again, essentially the first chapter of my thesis, which is focused on quantifying color pattern complexity and looking at some cool macroevolutionary patterns. And in this study specifically, we find at best weak evidence for the sexual selection idea, and especially after seeing the results of the path analysis, those effects are likely just mediated through body size, which indicates that these signals have more to do with detectability. So just to review, as small lizards are constrained by the size they can make their dewlap, they likely evolve these bold, high contrast patterns to increase detection. And large lizards, thanks. And large lizards, the conclusion isn't quite as simple. Of course, bold patterns such as this would be more detectable on a larger canvas. But the idea here is that these lizards aren't as constrained in how big they can make their dewlaps. So again, they have more leeway in what signal space they can exploit, and they could involve these more complex designs or intricate designs and still maintain detection. So finally, we also found this big difference between island and mainland species, that mainland species have more fine scale complexity and more color contrast than island species. And this big difference was not really well explained by some of the more proximate measures I was able to measure, like SSD and size. But again, it's likely due to things related to population structure like density and number of co-occurring species. So that's what I'm going to be focused on for the majority of the rest of my PhD. And as long as COVID doesn't get away with my fieldwork, again, I'll be going to the field, getting objective spec data for color in the light environment. Like I mentioned, data on things like community richness and density. And I'm also working on a parallel project modeling how these dewlap patterns differ at different viewing distances. So if you're interested in any of this, please stay tuned for future talks and papers. With that very fast talk, I'd like to thank all these amazing people who have been so helpful throughout my PhD so far, and all the people who have contributed to the many images that are needed for this macro study. And thank you guys for listening. I'll take any questions now.
Great talk. I don't see anything in the chat or Slack yet, so I'll ask my question, which is, it seemed like there's some interesting hypotheses, not just with the kind of directional patterns in these data, but also differences in the variation. Like, do those small species, are they more constrained, could be, or less heterogeneity in the data? But it seemed like from your figures, there's just a lot of variation in the things that you measured, and it seemed pretty uniform.
Yeah, there's a lot of variation. These things are so diverse in their patterns, and the biggest issue, or not issue, I guess, more challenge has been trying to quantify that information in a way that we can actually use. So there's definitely a lot of variation, and we're also looking at PC axes, right? So those are going to have a lot more noise in them than if you're just getting a perfect direct measure of that complexity, whatever that measure would be. If someone knows, please let me know. I'd love to talk about it.
Great, and there's a couple of questions. So let's see.
We've got time for one quick one, maybe.
Yeah, okay, I can answer any more on Slack or in the chat later.
Yeah, you'll have a bunch to answer. I'll put in the first one, Mariah's, which is awesome job. What about detection from predators? Is it possible that subspecies that experience heavier predation pressure can exploit all the trait space they might otherwise evolve?
Yeah, so in terms of predation, it's kind of funny with dewlaps because it is a signal that you can hide behaviorally. So that gives you a little bit more leeway to evolve a more detectable pattern. But on the other hand, they do display quite a bit. So the pattern's going to be out a lot. So one of the ideas about how these big dewlaps have these more intricate patterns, which are likely less detectable than the bold patterns, is that these big species are going to have higher predation pressure. So they might not want to be as detectable to predators. It's huge signal saying, hey, look at me, eat me, to all the birds and snakes that are watching.
I think we'll have to leave it there. Thank you, Jill. Sorry to interrupt, but
yeah, no problem.
Move on.
Thanks, everybody.
Our penultimate talk in this session is by Scott Taylor, titled The Genetic Basis of Spatial Cognition in a Food-Catching Bird. And I'll turn it over to Scott.
Awesome. Thanks, Howard. Can everyone hear me and see a single slide okay?
Yep.
Great. Well, it's nice to be with all of you, even though we're not together in California. It's a good reminder that there is cool work going on out there, regardless of how isolated we all feel. So today I'm going to talk about a really fun project that I've started recently with Vladimir Pravasudov's lab at the University of Nevada in Reno. And all focused on understanding the genetic basis of spatial cognition in birds that I think are among the cutest and most hardy of birds in North America, the chickadees, specifically mountain chickadees. So my collaborators on this project include Vladimir Pravasudov and previous students and current students in his lab, including Carrie Branch, Angela Pietra, and Ben Sonnenberg. They've done all of the field-based data collection of spatial cognition, so measuring spatial cognition in the field. And then two postdocs in my lab, Georgi Semenov and Dominic Wagner, have been the leads on the genetic components. So I think when many of us look at an image like this, we think of, powder days, skiing in the mountains, maybe. I mean, I realize not everyone likes skiing, but at any rate, this doesn't look like a really harsh environment for humans. The reality is for a lot of small endotherms that live in these environments that don't migrate, they need to overcome winter, and winter is a really harsh time to be in the mountains. And so for chickadees in particular, being able to recall locations of cached seeds is important for their overwinter survival. So chickadees are what we consider to be cache hoarding birds. So as winter approaches, they hide literally tens of thousands of seeds that they then recall the locations of during winter in order to survive. And we know from a lot of really amazing work that natural selection does shape variation in chickadee brains and subsequent cognitive function. And I'm going to unpack that a little bit, but based on work that Vladimir and others have done, we have a lot of evidence that natural selection is very, very strongly shaping these chickadee populations. And in particular, natural selection is shaping the hippocampus of these birds. And so the hippocampus is a region of the brain involved in spatial memory processing. And here I'm just showing you where it would be in a corvid. Hard to find sometimes in a tiny little chickadee brain, but a very important component in their ability to both, to recall where they've hidden their seeds. And based on work out of Vladimir's lab, we know that chickadees, both black-capped chickadees, in this case at higher latitudes, and mountain chickadees at higher elevations, have a larger hippocampus overall. They have more and larger hippocampal neurons. And as adults, they have a higher rate of hippocampal neurogenesis. So a lot is going on in the hippocampus of these birds. And when these birds exist in harsher environments, there is a difference in this region of the brain. And we also know, based on Vladimir's work, and I'll explain how we get these measurements in a few minutes, that chickadees in harsh environments, so again, either chickadees at high latitudes, you could consider black-capped chickadees compared between Ontario, Canada, and Alaska, or mountain chickadees at low and high elevations, the birds at these harsher environments, so these higher latitudes or higher elevations, have on average better spatial memory. So there's a physical difference in their brains, and then there's a phenotypic difference in their memory. And there's a lot more known about the Sierra Nevada mountain chickadees because of the number of years that Vladimir's team has been working on this system. So we also know that adult mountain chickadees in the Sierra Nevada have better spatial cognition than first-year birds. So if you average spatial cognition in adults, it's much higher than if you average spatial cognition in that first cohort, birds experiencing their first winter. Juveniles that have better spatial memory are much more likely to survive their first winter than juveniles that have poorer spatial memory. And I just want to say now too, people start, when I talk about this stuff, think about, okay, there's dumb chickadees and there's smart chickadees. But even the poorest performing chickadee on a spatial memory task is still has better spatial cognition than something like a chicken, for instance. So they're still high performers, they're just not as good. The survival of juveniles at higher elevations is lower than adult survival at high elevations. So again, there's this bottleneck that happens during the first winter where individuals below a certain threshold in a given year, keeping in mind that there's variation year to year in how harsh winter is, those juveniles are less likely to survive than adults that have already made it through that period of strong selection. And really importantly, cognitive performance, at least on the tasks that that Vladimir's lab is conducting, doesn't improve with age. So the cognitive performance that you have in the first winter as a juvenile is the same the next winter and the winter after and so on. And I think they have some birds where they have 4 years of cognitive testing and there's no significant difference across those years. And then finally, and I think pretty interestingly, females that are mated to males with better spatial cognition actually lay larger clutches. So even though cognition or spatial cognition isn't directly related to survival of chicks because it's the summertime when there's lots of food available, females are still cueing into this in some way and laying larger clutches when they mate to males with better spatial cognition. So I met Vladimir a few years ago when I was invited to give a talk at the University of Nevada, and we got thinking about, they have all this amazing evidence that there's strong natural selection on this trait. Obviously the trait is, well, not obviously, but most likely it's highly polygenic. And so could we use some of the methods that I've used, in my postdoc to look at, genetic relationships between phenotype and genotype on something as potentially polygenic as spatial cognition. And so we're leveraging whole genome sequencing and this amazing data set of spatial cognition collected in the field to try and understand what's going on at the level of the genome. So just to tell you a bit about the system, not too far from the University of Nevada in Reno, they have multiple field sites at different elevations. And so this, interestingly, it's not a huge elevation difference, but there's big differences between the low elevation indicated here by 1900 meters and 500 meters up at 2400 meters. And so they measure spatial cognition at both of these sites at low and high elevations. Importantly, there's no population genetic structure between these sites. So even though there's a strong gradient in natural selection where much harsher environments, many more feet of snow exist at the high elevation in the winter than the low elevation, there's no population genetic differentiation between these sites. And so to measure spatial cognitive variation, Vladimir has continuously refined these really amazing feeding arrays. And I'll zoom in on one in a minute, but the reason that they're suspended the way they are is because this is the way you can get bears to not destroy them. And in each array, there's 8 feeders. And the feeders can be set either as open, which means the little doors you can see are just open and a bird can come and land here and take a seed. They can be set as all, which means there's actually a door here that can open and close. And so if they're set as all, as long as a chickadee with a pit tag lands on this, which is an RFID reader, so a radio frequency reader, the door will open and they can get a seed. And they can also be set as target. And target is the really important component where once the birds have been trained to come to these and collect seeds, target means the door will only open for a specific bird. And so a trial starts, basically these feeders are set as all for 2 weeks. So the birds, any bird that comes that has a pit tag lands on the feeder, the door will open for it. And then once a trial starts, you will only open one feeder for a specific bird. And the way that spatial cognition is measured is the number of location errors a given bird makes. So as soon as a bird lands on any of these any of these RFID readers, the trial starts. And then the number of errors is the number of feeders, the number of incorrect feeders the bird goes to before it goes to the one that gives it actual food. And so what does that look like? Here you can see a pit tag bird collecting its single seed, and don't worry, the doors open and close at a slow enough rate that they can only get one seed and they don't get stuck. And then they fly away to cache it, but a pretty amazing system that he's developed. And so for the genetic component, or our component of this work, we collected genetic data from 42 individuals, and we sequenced whole genomes to a depth of about 15x coverage. And we used 2 different methods to try and understand if there was a genetic correlation between performance in this cognitive task and genetic variation in these birds. Keep in mind, there's no population genetic structure between high and low elevation, but there are ecological differences. And so we chose our individuals carefully, and I'll show you that in the next slide. And then we ran an admixture mapping program, genome-wide efficient mixed model associations, or the acronym for that is GEMMA or GEMMA. And then we also ran a random forest classification analysis, which is a type of machine learning, because in these potentially very polygenic traits, a typical GWAS might not be powerful enough, especially with the very small sample size that we're using. So this slide shows that we included high and low performing individuals from both elevations in our preliminary analyses. So low and high elevation are indicated by either green circles or purple triangles. And within each of those, there were high and low performing individuals. So by doing this, we hopefully got rid of any correlations with elevation, because we have low and high elevation individuals in both the best category and the worst category. Okay, so I'll just jump straight into the results. This is the results of the genome-wide association analysis, where we included sex as a covariate, and we used a continuous phenotype. So the total number of errors is the cognitive phenotype. Obviously, better performing birds conducting fewer errors. And so using 12 million SNPs across 42 individuals, you can see that a number of loci fall out above a significance value of negative log 10 alpha of 7, which is very, very significant. And even with a small sample size, we don't expect many of these to be false positives. And I'll talk about these a little bit more. When you use Bayesian sparse linear mixed models to estimate the number of loci, well, first we'll say the proportion of variation explained is quite high by the whole data set, and the proportion of variation explained by loci with large effect is also quite high. So it seems like there's a real biological signal here, which is exciting. And when you look at the number of potential loci involved in this spatial cognition trait, it seems to be around 10 with a standard deviation of 30. So a lot more than, you know, a single locus, but certainly not, it doesn't seem like it's hundreds of loci kind of trait, which is interesting. And I should say that all these plots just are arranged across the black-capped chickadee genome. The random forest classification analysis came with, produced 300 or 3,000 loci with positive variable importance scores. And so in these machine learning algorithms, if a locus has a positive variable importance score, then it has some degree of explanatory power associated with the phenotype you're looking at. And then you can subset this data set however you want. We ended up subsetting it by just taking the number of genes that appear in this set of 3,000 loci that have 2 or more hits, and then did some enrichment analyses. And so we ran enrichment analyses, so gene ontology enrichment analyses on these 2 data sets. And the results are really comparable between the genome-wide association, which had about 1,000 loci above that, or above the log7 alpha, and the number of loci that had 2 or more SNPs in the same gene for the machine learning analysis was about 1,500 as well. So when we run the Go enrichment analysis, we do see that these outliers in both analyses are enriched for regions of the genome involved in neuron growth and development, which is really exciting because, like I told you, we know these higher elevation birds or higher performing birds have larger hippocampus because they have more and larger neurons, and they also have a higher rate of adult neurogenesis. And all of these genetic associations and these overall enrichments agree with that. You can look at this a different way, and it's a little bit ugly for a slide, but if you just look at the highlighted red box, you can see that neuron differentiation, the generation of neurons, neurogenesis, and nervous system development are all overrepresented. And we can subset, you know, we can use different genomes to run these enrichment analyses, and we can subset our data set differently, but we always end up with really high enrichment, which is pretty exciting for what is probably, you know, it is a polygenic trait, but we're still finding these genetic associations. We also calculated FST between the best and worst performing males just to get rid of any potential sex variation. This is, again, a 12 million locus sliding window approach, but again, here only 24 individuals. And these 2 SNPs these 2 genes that I'm highlighting here, SSBP2 and CSNK1G3, are both the most strongly associated regions of the genome in the genome-wide association study, as well as FST outliers. And so it's not as cherry picking as it seems, although it still kind of is. But when we zoom in on these genes, we see that single stranded binding protein 2 is associated with adult neurogenesis in mice. And specifically, it's associated with neuron survival and number of neurons. And so this is a region of the genome that really differs between high and low performing birds. And then the other gene, casein kinase 1 gamma 3, it regulates fast synaptic transmission, which mediated by glutamate. And this gene has been shown, differential expression of this gene has been found in looking at the hippocampus of Alzheimer's patients. So even though the spatial cognition phenotype is obviously complex, and even though we only have a data set of 42 individuals, we're still finding genotype-phenotype associations that I think really excitingly are quite, I guess they're kind of obvious, but finding this it's still very exciting to me. You can imagine that for birds that are higher performing, you know, there's got to be really, really strong selection on these populations. And we know that in some chickadee populations, you know, 50 to 70% of a population can die in a severe winter. So I'm just going to wrap it up by saying, you know, these within population comparisons in mountain chickadees are providing what I think is really exciting insight into the genetic basis of spatial cognition. And the fact that we're finding that it's neuron growth and development, you know, keep in mind that we don't see this spatial cognition phenotype improving with age. It's got to be something to do with the substrate on which memories then are formed in the hippocampus, which is pretty awesome. So just to summarize, we find that there are quite a few loci associated with differences in spatial cognition in mountain chickadees, but maybe not as many as we would have expected. And we're really excited to up our sample size and really kind of dissect this and understand which loci are important and which ones maybe are false positives. But yeah, thank you. Overall, loci associated with spatial cognition in chickadees are in pathways related to neuron growth and development. And I think most excitingly, now we can start to examine short and long-term evolutionary dynamics of selection on this polygenic behavioral trait in the wild. And so we're planning on looking at allele frequency variation across the elevation transect in a larger number of individuals. Typically, they don't sample between these two sites, but this plot you're seeing here is snow depth. And so you can see a couple things in this plot. That one, it's annually very variable. So in 2015, very little snow. In 2017, over 400 centimeters or 4 meters of snow at the high elevation site. So there's temporally varying selection, which we can also use really cool new methods to examine. We have all the pieces we need for that. And we can also examine these strong selection events. So, how do allele frequencies change in the genes we've identified across these really strong selection events? And I'm really excited to start looking at that. And also really excitingly, we have another system of mountain chickadees right outside of my door. And you can see in my fake background at the Mountain Research Station at CU Boulder, where we're going to establish these testing arrays to look at the geographic consistency of the genetic basis of spatial cognition in these birds, which is super exciting. So with that, I'll just acknowledge my lab, Vladimir's lab, and all my friends and collaborators who have helped along the way. And I'll take any questions that anyone will have. Thanks.
Great talk. A lot of questions already in the chat. One, given this polygenic trait, how do you expect the variation to matter between individual populations of these chickadees, not just along the elevational gradient?
So how do you expect variation to matter between individual populations? Yeah, I'm not sure. I think it's going to be really interesting to see how geographically consistent, between the Sierra Nevada and between the Colorado Rockies, do we get outliers in the same genes? Obviously, because it's so polygenic, there's going to be multiple ways you can be a better performer or a worse performer. And we're at such an early stage, I don't even know what differences we might find. But certainly, I think possibly even within the Sierra Nevada, where these chickadee populations have been variously isolated and then re-expanded after glacial events, we might see population-specific variation in the trait. So it'll be really cool to kind of piece that together as we go forward. Unfortunately, getting the cognition data that you need is challenging, but if we get the link really tight, we can just start looking at allele frequency variation in those genes without the phenotype, just to see what's going on, which I think could be cool.
And I'll put one more quick one in there. Do you see any loci that are FST outliers that are potentially related to other aspects of adaptation to high elevation?
We do not. So because we included low and high elevation individuals in the best category and the worst category, we got rid of that variation. And even when we just look at low and high elevation individuals, there are no outliers, FST outliers. Even though there are going to be physiological differences, that's something we want to look into more.
Great. There's a couple more in the chat that maybe you
I'll get to them. Yep. Thank you, everyone.
Okay. Thank you, Scott. Last but not least, we have our final talk of the session. I'll remind our audience too, there's a couple of extra virtual talks available on the website you can watch at any point. But our final talk is by Diogo Provete. I believe it's going to be a recorded version, but he'll be here to answer questions. So hopefully we can get this one running smoothly.
I'll do my best.
Sorry, the title of the talk is The Tempo and Mode of Tadpole Body Shape Evolution.
Thanks everyone for watching my talk. This is the work done by my former master students, Marco Severini, and it's titled The Tempo and Mode of Tadpole Body Shape Evolution. So as we know, free-swimming tadpoles occur in a wide variety of habitats with distinct hydrodynamics and hydroperiods, such as permanent and temporary ponds, streams, phytotemata, and tree holes. Previous studies have also pointed out to repeated events of convergence in the morphospace of these larvae throughout their evolution. And these convergence events seem to be related to the hydrodynamics of each habitat, such as the contrasting between lentic and lotic habitats. And in this figure we can see that the amount and type of microhabitats available for tadpoles in ponds is much less than is much lower than those available for tadpoles in streams. They are much more kinds of microhabitats in streams. We also know that the morphological variation is much larger in larvae than in adults. However, little is known about how microevolutionary processes such as adaptation to novel habitats may have contributed to the production and maintenance of morphological variation in larvae. And we also don't know very well the role of habitats with contrasting microhabitat heterogeneity, such as ponds and streams, on the determination of variation in tadpole body shape evolution. So to wrap up this introduction, pond tadpoles have much less morphological diversity, and ponds offer much less ecological opportunities, while streams tadpoles have much higher morphological diversity and also offer much higher ecological opportunity because of the kinds of habitat available for tadpoles to occupy. And when we count the number of morphological guilds, we can see that the number of guilds is much higher in lotic than in lentic places, in lentic habitats. We also know that the ancestral habitat for frogs and tadpoles were ponds, were lentic habitats, while some lineages have invaded streams only recently in the history of the group. So with this work, we wanted to understand how this change in habitat may have altered both the rate of evolution and also the adaptive landscape for body shape. So we have a hypothesis for both the tempo and mode of evolution. We think because of this invasion of a new habitat, stream tadpoles will have much higher rate of evolution than pond species. And we also think that the adapted landscape will have separate peaks for ponds and stream species, while we expect to reject the null hypothesis with a single peak for both species. So to investigate this question, we collected data, morphometric data for 54 species from 11 families which occur in the whole Neotropics, using both landmarks and semi-landmarks in lateral and dorsal views. We took habitat data from the collection and the museum book and was coded as just lentic or lotic. We went through standard GPA, which is standard in geometric morphometrics, to process the data, and then we use recently published phylogeny to build a phylomorph space. To compare the rate of evolution between pond and streams, we use a method that assumes Brownian motion, and then uses a Monte Carlo procedure to generate a node distribution and obtain a p-value. We also used RI-Philo to estimate the rate of evolution in each node and tip of the phylogeny, and this method also automatically finds rate shifts, nodes with rate shifts in the phylogeny. To understand the adaptive landscape and the mode of evolution, we use phylogenetic EEM, which is a multi-peak OU model that estimates the peaks, the adaptive peaks, and also search for peak shifts. For this analysis, we only use the first two principal components for shapes. And what we found was an evidence for convergence. So there's lots of both streams and pond tadpoles in the center of the phylogeny, in the center of the phylomorph space. This is for lateral view. but we also found some evidence for directional evolution in some lineages, such as Psychorhynchidae, and also evidence for clade disparity, because the amount of the the size of the phylomorphic space occupied by stream tadpoles is different from the pond tadpoles. This pattern was repeated for dorsal view, which also highlighted microhelid, which is occupies a quite distinct region of the phylomorphic space. The rate of evolution between stream and pond tadpoles were quite different, with stream tadpoles have much higher rates of evolution, and this difference being significant. And we also found evidence for two rate shifts in lateral and also dorsal view, two lineages. As for the adaptive landscape, the best model that described the data found that there were 6 optima for lateral view and 4 optima for dorsal view. So our results support the initial hypothesis that lineages from lentic habitats which invaded recently lotic habitat had a different rate of evolution and also shared distinct optima in the adaptive landscape. Some lineages also seem to follow directional evolution in the morphic space, but there was also clade overlap between species from lentic and lotic habitats, and also clade disparity, because species from ponds and in streams occupied different region in the phylomorphic space. And the rate of evolution from pond tadpoles was much lower than that of streams in both views, but there is high heterogeneity in rate within each habitat. As for clade overlap between species from pond and streams, the mechanism that may have formed this clade overlap was the mode of evolution, mode of speciation of species, which may have contributed to the morphological similarity, but also the similarity in terms of hydrodynamics of pools inside streams and ponds may also have exerted similar selective pressures in these tadpoles. And as for the directional evolution of a family, Psychorhynchidae, so these tadpoles occupy extreme positions in the morphic space, and also they also occupy microhabitats, which is wetted rock surfaces in streams that requires a specific adaptation to attach to rocks. So this is kind of stream habitats may have accelerated the rate of evolution as a way to adapt to this novel habitat. And as I said, frogs only invaded recently streams, and streams are also much more advantageous in terms of overposition sites, because they have much less predators and competitor density, and also have stable hydro periods, while ponds have variable hydro periods and much more predators and competitor density. However, they also offer a challenge in terms of hydrodynamics, because to which frogs have to adapt. And because of phenotypic plasticity, larvae can respond in different ways to these new development sites, which may have contributed to the creation of a higher morphological diversity. So we think the plasticity first hypothesis as a way to create new novel morphological novel forms may be the main mechanism explaining the formation of this new, the fixation of these new phenotypes in this kind of habitat. Streams also offer another challenge, which is its low primary productivity. Ponds have much higher primary productivity, which allow tadpoles to stay much less time in ponds than in streams. So this longer development time in streams may have contributed to alter the timing of developmental events, which is called heterochrony. And this developmental mechanism may have contributed to the formation of of new evolutionary novelties. And as the formation of, as a hypothesis for the formation of the adaptive landscape, shifting balance theory may explain the creation of new peaks, because well, this theory states that populations may go to new peaks when they have low effective population size coupled with drift and reduced fitness, and during this decreased times of decreased population size, populations with reduced fitness could go to new peaks, adaptive peaks. And stream frog populations have considerably low population size than ponds, so this may have contributed for those to go to new peaks and with increased population size, and then which will go through natural selection. So I want to thank CAPES for funding and all the curators who provided access to collection under their care. And if you have questions, feel free to type them in the Slack channel or just email me directly. Thank you.
Great talk to end the session, and there's already been some questions answered in the chat about controlling for tadpole stage. If there's any more, I'm happy to relay them.
I guess that's an advantage of a recorded talk, is you can answer slides you can answer questions during the talk. Well, I'll take the opportunity to thank all our speakers for some very interesting talks in the session today, as well as our technical and question hosts. The things went reasonably smoothly. Hopefully this is the last time we have to do this as a virtual Asilomar. Looks like people are end of a long day and starting to head out. So thanks everyone for coming, and have the good rest of the weekend and rest of the conference, hopefully.
Also a reminder that later this evening, there's a plenary by Scott Edwards. So it starts at 9 Eastern or 6 Pacific. Should be really good.
Thanks for that reminder, yes. Okay, take care, everyone.